Dear colleagues, our session is going to start in 4 minutes. Okay.
So it's time, yeah?
Yeah.
Dear colleagues, my name is Oleg Kotsurvenko. It's always my pleasure to lead the astrobiology section at the annual conference at the Space Research Institute. Now we have a lot of interesting talks. Let's start with the first presentation. It's Vladimir Kompanichenko with the talk concept of thermodynamic inversion as a model of the origin of life on planets and satellites. So please, Vladimir, you can share your screen.
Vladimir, we see your screen, but we don't hear you. Could you please unmute yourself? Vladimir, do you hear us? You are muted.
Vladimir, can you unmute your microphone? Oh. Yeah, now it's perfect.
Is it okay now?
Now it's okay, yeah.
Yeah.
Okay. In my presentation, I will express key notions of the concept of thermodynamic inversion, shortly TI concept, along with some consequences for astrobiology. This is a basic picture with illustrating the barrier between non-living chemical systems and living biological systems. Below this border, there are three main types of prebiotic models: RNA world, protein world, and lipid world. And above this border, there is a root of a phylogenetic tree of life on Earth. As an example, let's consider one of prebiotic models suggested by some Japanese scientists. This is lipid vesicle along with incorporated DNA. Next step of evolution is amplification of DNA through the temperature changes, and the last step is division of the lipid vesicle along with amplified DNA. The same process is shown in pictures, and after that, further evolution of this model stops. So, this model, as well as all other types of prebiotic models, are unable to evolve further to life because they do not possess the following key properties that demonstrate activity of living systems in the environment. First, the ability to boundless expansion, and second, purposeful behavior, or the same initial sparks of foresight. So there is a gap between even most complex prebiotic systems and the simplest living cells. And this gap was illustrated in the picture by the same Japanese scientists. On the left, main types of prebiotic microsystem, on the right, living cells. And this is gap between them. Let's consider this gap, coming back to the initial picture from the point of view of chemistry and thermodynamics. In the chemical context, chemical evolution and further biological evolution proceed in the same direction, that is, with continuous complexification of organic matter. And in this context, biological evolution looks like a natural continuation of chemical evolution. However, from the point of view of thermodynamics, this the situation is quite different. We know of the second law of thermodynamics that any non-living system exists or evolve with continuous increase in entropy, while biological evolution of biosphere, proceeds in the opposite thermodynamic direction with accumulation of free energy and information. The both tendencies are well known. Let's consider them, express this difference through the balance between the total contribution of entropy to the total contribution of free energy and information. It follows that there exists a principal gap thermodynamic gap between the non-living and living systems, because the balance in non-living systems is prevalent, entropy is prevalent, while in biological systems this balance is negative. On the contrary, free energy is prevalent here. So there exists a negentropic barrier between the non-living and living systems that manifests itself in the existence of a natural tendency of non-living systems to increase entropy in accordance with the second law, which makes it impossible for organic systems to spontaneously transit to life due to their chemical complexification only. This requires a thermodynamic inversion of the system, the entry into it of a huge negotropic impulse, which compensates this tendency in increase of entropy, compensates it in plenty. It is shown again in this picture, this initial prebiotic microsystem, this tendency to increase in entropy, and in case it obtains a huge impulse of free energy that compensates this tendency in plenty, for a certain period of time, the systems rise above the negentropic barrier and exist in the thermodynamically inversed state, that is, with free energy contribution prevalent over the entropy contribution. In the long run, the system coming back to the initial passive state due to natural increase in entropy in accordance with the second law. And just this initial impulse of free energy can occur under strong and optimal changes in the environment. And in the case the environment is oscillating with oscillation of thermodynamic parameters, temperature, pressure, and so on, the system begins to, from time to time, to exceed the negentropic barrier and begin to oscillate around this barrier. And two possible trends of evolution from here, from the intermediate state between life and non-life. These tendencies first to biological evolution with the continuous increase in free energy surplus by means of stepwise activation, or the full degradation of a system with the irreversible increase in entropy. Now, this tendency are shown in this picture. This is the initial state of intermediate state between life and non-life. This balance between free energy and entropy oscillates around the negentropic barrier, and two possible trends of evolution with rise in free energy through the stepwise activation or full degradation of these systems and its returning into the initial passive state, prebiotic state. So there are four key points of the concept of thermodynamic inversion, shortly TI concept. First, starting point, the intermediate state between non-life and life. Second, two thermodynamic trends of evolution, to active and passive existence. Third, increase in free energy levels occur through the stepwise activation. And the fourth, transition to life was stimulated by effective stress response to external oscillations. And the how these four key points outlined theoretically we can find in real existence of microorganisms and their populations, mostly in the anabiotic process, that is transition of a living cell, microbial cell, to the anabiotic state and its exit and return to the active existence again. The both ways of the transition are universal. All the time, the transition to anabiotic state due to change external conditions begins with the increase on functional activity of the most complex structures, and it completes with when the cytoplasm acquires a glassy state. This is a state of anabiosis. And the exit to the active state again begins with the action of stress factor and begins with weak increase in respiratory activity and complex restoration of activity of genetic apparatus. So we can find here the whole 4 points that we outlined earlier. 2 trends to active and passive existence, the intermediate state between non-life and life, when the S cell is in the state of anabiosis. Exit begins with impact of stress factors, and the process of exit is proceeds through the stepwise activation. This correlation between theory of the TI concept and facts, real facts from the theory of anabiosis are shown here, and it is marked in red. And the following thesis is put forward. Since the transition of a bacterial cell from passive to active state always occurs through the same sequence of changes in metabolic processes, it can be assumed that this sequence itself arose in the earliest period of the emergence of primary life form. And this sequence has been repeating itself during the entire evolution of biosphere. And this means that the stages of transformation of prebiotic microsystems into primary life forms had been recorded in the stages of the exit of a bacterial cell from anabiosis. Just this succession from the initial intermediate state between life and non-life, just to full exit of a resting cell from the anabiotic state. And using this universal succession of the exit of a bacterial cell from anabiosis, we can reconstruct the succession of steps of the origin of life on the early Earth, integrating this succession with the geological environment. There are 4 main required conditions for the origin of life, 3 accepted ones, and I add one more: multi-level oscillation of physicochemical parameters in highly non-equilibrium medium. From this point of view, hydrothermal systems such conditions are usual for hydrothermal systems with high fluctuation of parameters, while such oscillations are absent or almost absent in ocean. There is one of examples of oscillating multi-level oscillations of pressure of fluid in Kamchatka, irregular macro oscillations and regular macro oscillation with a period of about 20 minutes. And the 4 stages of the origin of life. On the right, geological conditions, stages of transition in the course of pulsating migration of hydrothermal fluid to the surface. First step, self-assembly of prebiotic microsystems, non-living, under such conditions. Second, formation of protocells, which are in the intermediate state between non-life and life, appearance of weak energy-giving process of respiration, and so on. Third, formation of primary living subcells, appeared the protein synthesizing apparatus and primary RNA templates, and the last 4th stage, formation of progenotes with the full growth cell cycle and the formation of the genetic apparatus. And these stages are shown in this picture. Self-assembly of non-living prebiotic microsystems, intermediate state between life and non-life, appearance, rising of RNA vault, formation of progenotes, and further evolution up to prokaryotic cells. And within the hydrothermal channel, this process stimulated with high frequency fluctuations of physicochemical parameters, while the near surface conditions, radiation may also contribute into as a stimulating action for further stimulation of the origin of life. And 3 main consequences. First, the general approach outlined here can be applicable for other planets because they use thermodynamic notions, free energy, information, and entropy are common for the universe. So, simple life should be a widespread phenomenon in the universe. Second, life could have originated on some bodies of the solar system, since at least Mars, Enceladus, and Europa there are signs of all 4 required conditions for life emergence: water, organics, energy, and fluctuations. Third, search for microbial life beyond Earth should be aimed at finding the traces of both, first, actual prokaryotic cells, life as we know it, and second, simply microsystems whose evolution stopped at the proto-life level. I mean these stages. This is my acknowledgements, and summary, you can read it by yourself. Thank you for your attention.
Thank you very much, Vladimir, for your interesting presentation. Now we have time for questions.
We have one question here.
Yes.
Nikki.
I have a rather standard question. When you are talking about 3 conditions to arising of life, usually there is a 4th condition, a stability of this system. We need some time to form life. And you did not mention anything about the time duration or some stability conditions of the as you call, prebiotic microsystems. Yes? What about the stability of such systems and what time they should?
Okay, I understand you. Multi-level oscillations of parameters are necessary to maintain oscillations of prebiotic microsystems around the negentropy barrier, without oscillations, this process is impossible, because any transit of a microsystem over the negentropic barrier needs huge impulse of energy, and such impulse can be obtained only from the medium with high fluctuations parameters. But you are right, these parameters should oscillate in optimal regime, optimal mode. In this presentation, I didn't consider my another scheme of the origin of life under far from equilibrium conditions following Ilya Prigozhin. Under such condition, when the system transits from initial stable state to the advanced stable state through the point of bifurcation, the optimal position of the microsystem to evolve further to life is between two these attractors, initial stable state and advanced state. So to keep this stability between these two opposite trends is possible under small oscillations. So life started from paradoxical, stable and simultaneously unstable state. Instability is oscillation of parameters, and stability is equal attraction from the initial and the advanced state. I'm sorry that long speech here, but I should tell this. So combination of strong changes and weak stages during the origin of life process is necessary. Thank you.
Thank you, Vladimir. More questions?
I will. One more question. I also have some general question. No, it's very important topic you discuss, but thermodynamics is a sort of average, very general description of the processes. And as I understand you, and I agree with you, what you describe as necessary conditions for transition from to life. But what is but it still remains mystical, even in your description, because the moment of transition, what's the mechanism of it? Thermodynamics is good, but it could not describe the particular mechanism. Do you agree with me that you describe necessary conditions, but not sufficient conditions?
If I understand your question correctly, maybe these four required conditions maybe can be insufficient and needs maybe one more conditions that we don't know now. Maybe. Is it my response to your question is right? Can I have I understood your question correctly?
Approximately, yes. You agree with me that it's necessary conditions, but you need something else, maybe something mystical, maybe the God presence, or I don't know what.
I absolutely agree with you. We need four required conditions and something more, and that we feel, that I feel, but we cannot formulate now. I cannot formulate now. Something more. But now only four conditions I can formulate.
Okay, we have another question. Yes.
You know that now there are a lot of publications related with the production of hydrocarbons, nitriles in the atmosphere of Titan. And moreover, in this atmosphere, there are a liquid phase as methane lakes. How do you think there are conditions for the any step of this life production in these conditions with very low temperature, but with a lot of atoms of hydrogen, nitrogen, and carbon, and with very small quantity of oxygen?
In my opinion, high temperature is necessary for the origin of life, and optimal chemical compositions of prebiotic microsystems is insufficient to transit to life. In the temperature of about below than minus 100 degrees is too cold temperature to form life. But maybe earlier Titan was possessed high temperature, and such transition might occur. And this is one aspect. Another aspect is I talked about life as we know it, but maybe we can find another kind of living systems on Titan, as suggested by Chris McKay, for instance. Maybe my main point is that during the origin of life, the balance between entropy and free energy all the time displaced into the more and more prevalence of free energy and the entropy. So the more and more surplus of free energy and in comparison with entropy. So maybe such a process took place in Titan and used another chemical composition to redistribute organic systems and water between cells and its environment.
Thank you.
Vladimir, in continuation to the previous question, since you're referring to the origin of life on planets and satellites, of course, there are a lot of questions about origin of life, extraterrestrial life, yeah, and of course, for me it's interesting, how can you explain maybe some specificity of such origin of life, for example, in Venusian clouds, not in the surface? Is it possible to apply your theory to this interesting system?
Yes, it is possible. We consider such possibility. There was with you, with our common paper, and the paper with some other scientists. It was possible in case the temperature, near surface temperature on Venus was below that 200 degrees centigrade. Under such conditions, it would be possible.
In clouds themselves, yeah? In this area of space. That's interesting. Not on the surface.
I don't know. Theoretically, it is possible, but it depends on how oscillating conditions would be suitable for such a process. Conditions in the atmosphere of Venus. In case very strong but optimal changes in the parameters, maybe such process could occur in Venus.
Okay, thank you. More questions?
We don't
Okay.
have more questions. We don't
Okay.
have more questions.
Do we have?
No?
No questions. Okay. Thank you very much, Vladimir, for your interesting presentation. And now we
Thank
go to the second speaker. This is Valery Shematovich with his talk, Non-thermal nitric oxide formation in polar regions of nitrogen-oxygen atmospheres.
Good morning, dear colleagues. Our talk is more prosaic, but nevertheless it's is connected to the astrobiology problems. I would like to talk about the non-thermal nitric oxide formation in polar regions of N2O2 atmospheres, and how this nitric oxide, this minor species, could be connected to the fact that we have life on our planet. This study was conducted together with Dmitry Bisikala and Grigory Tsurikov from the Institute of Astronomy. Some few introductory slides. We have three rocky planets. In reality 4, but with atmospheres 3 rocky planets in our solar system, and you can see how their atmospheres are different. Looking on Venus, Mars, and Earth. And Earth's atmosphere, and Earth, we have one planet with a life, and we see what is the difference of atmosphere on Earth. It's a N2O2 atmosphere, and usually in astrobiology the main point of view and main interest is connected with the formation of molecular oxygen and so on. But recently the attention was paid to the formation of why N2 is the main component of this atmosphere and how it determines and constrains the formation of life on our planet. And probably there was a paper in 2019 by Helmut Lammer and collaborators, and they argued that N2 dominated atmosphere in combination with O2 on Earth-like planets within circumstellar habitable zones can be considered as a geobias-signature, and there are some because N2 is formed mainly due to some aerobic biosphere. N2 becomes the main component of atmosphere, also due to the formation of life on our planet. And in case of N2O2 atmospheres, we see what species can be considered as some indication of life on such kind of planet. And N2 and O2 are both homonuclear molecules, and not many transitions and so on, and it's very difficult to observe these molecules, especially for exoplanets, especially for the space. Therefore, some additional molecules are suggested to be indicators of life on a planet or some biological activity. Usually it's molecular oxygen and ozone. But now, from the studies of different bodies in our solar system and in planetary systems around other stars, we know that O2 oxygen ozone atmospheres can be formed, and rather efficiently formed, without any biology. It's some photochemical sources and so on. Therefore, we propose to take into account, when you consider some biosignatures, both species, atomic nitrogen and oxygen. And one of such species, in our opinion, could be nitric oxide, because it's formed in N2O2 atmospheres rather efficiently, and it could be observed both in UV, gamma bands of nitric oxide, and in infrared, and space telescopes such as the Spectrum UV, Spectrum UV, WEF, and James Webb Space Telescope could provide some additional information on nitric oxide. But it's as a first step, it's necessary to understand how efficiently nitric oxide is produced in such N2O2 atmospheres. And from studies of the Earth atmosphere, we have some very important background in such studies, because we have our atmosphere and we have a lot of information about our atmosphere. And for example, you can see here measurements of nitric oxide in the Earth atmosphere. It's nitric oxide is formed in very narrow region near in a lower thermosphere, and it was interesting from this observation that there are different sources. There is photochemical sources in equatorial regions, and some precipitation of energetic particles in polar regions. And you see that the production rate in polar regions is a few times higher than in mid-latitudes. And therefore, we made this some kind of steps to understand and to solve this problem. We have some ideas that N2O2 atmospheres, it's a very important feature of planets, of rocky planets, and we can suspect formation of life, rising of life in such system. And after that, we are trying to find some set of biomarkers. And to do this, we started our study step by step, first to understand how nitric oxide is produced in different regions of planetary atmosphere, and how we can estimate the abundance of this molecule. And is it possible to see using a current and future space telescope, and to connect with N2O2 atmospheres and suspect this planet, rocky planet, that there are some conditions for a rising of life. Nitric oxide, it's a very interesting system because it's formed in a very narrow, very limited set of reactions. There are only 3 main reactions, and this system, all reaction rates are rather well known, and therefore, this system is very stiff from chemical point of view. If you slightly change one of rate reaction and so on, production is changed very strongly. And many years ago, we were involved in solving this problem, and we proposed to take into account some additional small reaction with, in which nitrogen, atomic nitrogen with the excess kinetic energy, reaction of nitrogen with excess kinetic energy could be taken into account to extend this system, because this reaction has an energy barrier, and for usual thermospheric temperatures, usually it's a rather low rate for this reaction. And okay, as a first step, we considered, in our opinion, very important source. It's a precipitation of high energy electrons in the polar regions of the Earth's atmosphere, and formation of nitrogen atoms with the excess energy in dissociation collisions. And we calculated energy distribution function, production rate, and energy spectra of this production rate. And you can see this form, and you see that it's totally non-equilibrium. Therefore, to follow the fate of or kinetics and transport of hot nitrogen atoms, it's necessary to solve Boltzmann equation. And we have such experience and we have such codes. And I would like to show the results of calculation of distribution of suprathermal nitrogen atoms in the S-polar atmosphere and how, and their input in the production nitric oxide. And here are the energy distribution functions, result of calculation of kinetic Boltzmann equation. We considered 3 test cases, with different characteristic energies of precipitating electrons. And you see, and the energy influx in the system was the same, 1 erg per square centimeter per second. And we see that in the region where nitric oxide is mainly produced, about 110 kilometers in the S-polar atmosphere, energy distribution functions are very non-equilibrium for nitric hot nitrogen atoms. Here, dashed lines is local equilibrium distributions, and solid lines is calculated. And you see, we start this from a reaction barrier, 0.3 electron volts, and we see that probably this nitrogen, hot nitrogen, could be some additional source for production of nitric oxide. Here we show the height distribution of these nitrogen atoms, and only one feature I would like to point out about our numerical model, because it's a first model in which we consider correctly transport of these hot nitrogen atoms. And when we calculated the reactions with the hot nitrogen, it's a black lines in these figures. You see that the input of hot nitrogen reaction with molecular oxygen is minor, but the system is stiff. These are still main reactions, 2 and 3 with atmospheric nitrogen atoms, but the system very stiff, and therefore when we calculated height profiles using some diffusion models of nitric oxide formation and distribution in the S-polar atmosphere, you can see that black line is a standard solution for the thermal energies. And when we take into account precipitation and with some characteristic energies, and we see that the densities are at the peak densities are strongly increased. And for example, when we have this very hot energy of 5 kilo electron volts, increase in peak value is about 30 times higher than for thermal case. And here we calculated column densities. This table shows values of column densities, and we know that from accompanying study by Zurikov and Bisikala that to be sure that nitric oxide could be observed with, for example, spectre UV, spectre UF space telescope in gamma bands, it's necessary to have something like column densities 10 to the 16, 10 to the 17. And we see that addition of rather strong precipitation, of electron precipitation, provides us such values. But it's only first step, because we have polar sources, and after that we should develop a circulation model and to see how nitric oxide is distributed around the planet, and after that only we can provide final estimates of possibility to see this molecule as an indicator of geo-bioactivity in a N2O2 atmosphere of a rocky planet, around probably around other stars and so on. Thank you for your attention.
Thank you very much, Valery. We have time for questions.
We have one question.
Okay.
Thank you for your nice presentation, but one question. When we consider N2O2 planetary atmospheres, we must take into account all mechanisms of the production and no productions. You have considered the interactions of excited and unexcited atomic nitrogen with O2. But about 50 years ago, Galeev, Sagdeev, Shapir, and Shevchenko have developed the theory of beam-plasma interaction.
Yeah.
And after that, Yevgeny Mishin, Rushin, and Telekin have applied this theory in the production of thin layers of NO in polar ionosphere. And they have applied the reaction of vibrationally excited molecular nitrogen with atomic oxygen. Because of the production of energetic tails in electron energy distributions. How do you think, is it possible the contributions of these reactions in this planetary atmosphere or not?
I see. Yes, thank you for this question. You know, you mentioned this another reaction of atomic oxygen collisions with the nitrogen vibrationally excited. It's a cycle, so-called, of Zeldovich cycle of nitrogen detonation or something like. But this reaction has a very, very high energy barrier, about 2.5 electron volts. And in thermospheric, sorry? But the vibrational levels are only less than 1 electron volt. They do not provide some this additional energy to overcome this energy barrier. But for reaction of N with O2, there is also energy barrier, but only 0.3 electron volts. Therefore, this excess kinetic energy allows to
have considered, they take into account vibrationally excited to 12 levels and higher vibrationals because of productions during the beam plasma interactions of energetic tails in Maxwell distributions of electrons.
I see. Yes, but you know, it's a lower thermosphere. It's a surrounding gas temperature is about 3 or 200 kelvins, you know. It's impossible to pump nitrogen, molecular nitrogen to this very high. Yeah.
Thank you. More questions, or there is an announcement?
Valery, no, two points. First one, it's very nice modeling, but it's intrinsically assumed that the planet has magnetic field if you have this auroral type precipitation. But nobody knows. We just did the study if trying to apply these ideas to exoplanets, but all people working on Dynamo could not say what are the necessary conditions for generation of magnetic field. We may have the large planets like Earth and Venus, and the situation will be very different. And then I think it makes sense to analyze also for exoplanets a Venus type of interaction where there is no auroral access. And second is a sort of Gedanken experiment. You describe this N2O2 as some signatures for possible life. But do you think what's happened?
Precipitation of electrons, we should this source of nitric oxide, for example, for such systems to take into account. But you are right that it's necessary to have proper ratio between N2 and O2 molecules. It's still open question.
Thank you.
Thank you, Valery. We have time only for one last question. This is Evgeny. Yeah, please.
Evgeny,
Thank you very much. I just have further comment on Lev Zeleny's question with regard to truly oxygen atmosphere. In any occasion of wildfire, we will get far that there is no way to stop in fully oxygen atmosphere. So even when there are any problems with raising of life in this atmosphere. Just any accident wildfire will burn everything to the ground. Thank you.
Thank you very much,
Thank you.
Valery, for your.
Could be existing on Europa, Enceladus, as shown here, on icy moons where under the ice sheet there is some ocean in contact with the rocks. And my comments are, if you find out the life there, it should be unique, of course, unique for every system. It means the life we could find out in Lake Vostok will be different, of course, from Enceladus ones and Europa one. And just two slides about Enceladus, because Enceladus is the best model now for searching the life. Theoretical model, because no missions there still sent. But you see here there is a water, there is some chemicals, and even very complex organic molecules with masses more than 200 atomic mass units. So Enceladus is the best candidate to find out extraterrestrial life in our solar system. And what is interesting, just recently James Webb Telescope detected that the plumes from Enceladus can be very huge, gigantic, I would say. And it was observed at 10,000 kilometers apart from Enceladus. So it's not needed to fly to Enceladus to land on Enceladus to find the life, if it's possible to pick up, to collect some materials from the plume. My talk is about Antarctica, searching unknown life in Antarctica. And the target is freshwater Lake Vostok, subglacial Lake Vostok. And I will talk about the accretion natural ice which originated from the lake water. But just a few slides to remind you that under the Antarctic ice sheet there is plenty of not lakes but water reservoirs, and the Lake Vostok is the biggest one, this one. And this lake is visible from the space. This is a radar set image. This is a Vostok station, Russian Vostok station. All picture, because now it's under snow and new station is now constructed, nearly finished. It's far from the coastal area. There at the surface, the temperature is very minus. Absolute minimum was detected there, minus 55 at the surface, but beneath the station there is a huge lake, Lake Vostok, and Russians started to drill toward the lake in 1990. The drilling operation was performed not for biology, of course, for paleoclimate, and together with French colleagues, 4 big interglacial cycles were detected there. But then, after the cycles were detected, the borehole entered so-called lake ice, озерный лед in Russian. It means there is no any climatic signal. In French, colleagues told us that this is, of course, water, just water frozen. So, Lake Vostok settings, just 2 slides. It's a huge, of course, lake. You see here the dimension, the total area, more than 6,000 cubic kilometers, 2 basins, and so on. What is important? It's buried under the 4 kilometers ice sheet, thick, and isolated from the surface biota for at least 14 million years ago, because Antarctica as a continent was moving from the equatorial area to the South Pole started to freeze. And no hydrological links until now is shown or known to other in connecting other lakes. And regarding biology, possible biology, the lake is a rather friendly environment for life because there is a free water, of course, fresh free water, but deeply buried. It's okay for life. Higher pressure, 400 atmosphere. It's okay. Not very cold. You see here, -2, -3 degrees of Celsius. But of course, no carbon, no organic carbon in the water. It's detected, it's analyzed. The ice was analyzed and no any organic carbon was found there. But we know that there is some microbes which can exist with no organic carbon, so-called hematite autotrophic life. But the constraints for the life is oxygen content on the lake. It's considered around this latest data here, around 800 milligram per liter of dissolved oxygen. It's a huge. Chris McKay sometimes told us that like a cold oxygen reactor. It's hard to imagine that the life could be existing there. Just a few slides where we treat the ice. It should be decontaminated, of course, because it was treated not for biology. And the dust-free room where the ice should be melted, water or material should be concentrated, and we fish out all possible DNA or molecule, including viruses, from this mix using these special membranes there. And in addition, we know the ice was treated not for biology. It's highly contaminated at the surface, and if you treat even the surface carefully, still some contaminant can left on your hands, on material, on combinations, and so on. And so we established our own contaminant database, which now include more than 300 species, these phylotypes or species of bacteria. Why? If you found something new, we just compare with this library contamination database and decide is it a new one or contamination. This is a very important message. We believe because of conditions unusual, except unusual in terms of oxygen, life on our Earth could not be adapted to such higher oxygen tension. Still not measured, of course, but it's calculated independently by Chris McKay and Vladimir Lipinkov from Archaeoeconomic Institution. And we believe that what we found out, it should be very special stuff, like extraterrestrial stuff, unique stuff, life form we have never encountered before. But this life should be adapted or quickly evolved to lake conditions which radically but gradually changed during last 14 million years. Because the lake area started to freeze, and the ice sheet started to grow. Conditions changed. Sometimes may ask me, 14 million years, it's nothing in terms of geology or biology. But I just would like to give you example. Evolution, in fact, can occur very quickly. Just 1 week ago in Nature appeared the paper. It depends on the circumstances. And in the lake Victoria in Africa, 50 species of fishes appeared in less than 70,000 years because of higher adaptability of these species and breeding. But bacteria also have very high potential adaptability and breeding, of course, via horizontal gene transfer. It means that then in a few dozen years you can get a species, but this is animal, not animal mom. No, I mean fishes. What about bacteria? Bacteria could be even in a short time to be adapted. And just the scheme how it was the drilling, and they reached the borehole, the lake, at the depth 3775. And I just show you the result. My presentation is focused on thermophile. Why is thermophile? Because when you work on ice, it's hard to imagine that thermophilic bacteria could contaminate your ice because they are thermophilic. They should live in a specific environment. It's not a contamination. Because other stuff, we found some other stuff, but it's problematic because this stuff, normal stuff, it could be there, could be in the lake, but thermophiles no, because they have a special eco-niches to live there. And I just show you our previous finding. It was thermophiles in Lake Vostok. It was published long ago, not 20 years ago, in fact, 2004, 2006. That time we found thermophilic hema-allelotrophic facultative microorganism, so-called hydrogenophilus thermolitolus. And when we found this in the ice, it's not contamination. Many other studies confirm that around the lake there is a special geothermal system where this thermophilic bacteria can exist. Not in the water, of course, which is -3, -2. And what is interesting, that bacteria, hydrogenophilus, was found in 2 different ice horizons, lake ice, accretion ice, natural ice, in 2 horizons separated by 5,000 years. So it's not a seldom finding, and we were happy because after that, I don't know. There is no scheme, I am sorry. Because after that it was decided that around the lake there is a special geothermal environment where this thermophilic bacteria can be living. And now we switch it to higher throughput 3rd generation sequencing technology, called Oxford Nanopore. Before the data was obtained using classical Sanger technique. It means not more than 100 clones. This technique, 3rd generation, can generate you millions of reads, millions of sequences. And we decided just to retest, re-evaluate those findings which were done around 20 years ago. Just show you the picture. It's a very progressive technology, Oxford Nanopore. It looks like this is a device called MinION. It connected to the computer, and you in real time the sequencing is run, but not for 1 hour, of course, maximum for 72 hours, 3 days, but in 2 hours you can get already some results. This is a special technique which use the property of special nanopores, protein nanopores, through which one strand of DNA passes, and the current changes in the current are recorded and then de-recorded to the nucleotides. And the results, thermophile re-evaluation, in fact. Just to remind you, that time Sanger, 20 years ago, we analyzed 1, 2, 3 samples of ice. The same horizon, 3707, but 3 different segments. In one segment we found nothing, in another one nothing, and only in these segments, they are all contaminants of course were present. We found 12 clones and so on. We found this stuff, Hydrogenophilus thermolitolus, in one segment of the same depth, but the depth is 1 meter, of course. And I told you 1 centimeter is very aged, I mean, because ice is pressed and the rate of accretion is 1 centimeter per year. And now, nanopore, we again come back to the same samples because we have archive of ice. We again took the same sample, 3707, where Hydrogenophilus 20 years ago was detected. And along this, we took another one, 08, 07 another segment, 07 another segment, and 09, just for comparison. And run nanopore sequence. I just show you the picture how ice looks like. This is 3607. There is a small inclusion inside, but this is a big inclusion visible by eyes. It's unique, the so-called big камина in Russian, большая камина or Grand Rocky in English. It's a unique example which monocrystalline ice, this is monocrystalline ice of lake water, contains inside the inclusion, mineral inclusion. And the results were astonishing. Why? We obtained more than 1 million reads, like clones, not 100, but more than 1 million. And we found the signal only in one, 3708, большая камина, as I told you. And for this sample, we analyzed more than 1,600,000 reads, and you see, and we found their so-called moderate thermophile. The name is Meothermus hypogeus. And we found not just one reads, because one reads or two reads, it's not statistically. We found 280 reads. It means it's a population of this stuff. And in addition to this, we found 27 reads for another species of the same genus, thermophile. This is just data to show how it's done. It's very complicated. You see here average score, average sequence length, how many were classified, unclassified, and so on. In total, thermophile was present in the sample only 0.02%. That 280 reads, because we found in total we analyzed 1.5 million reads, of course. So if we used censure technique, it was not possible in principle to recover this thermophile because it's a very low score of this. And therefore, thus, our new finding, it's astonishing. We found new thermophile of another Hydrogenophilus was of beta proteobacteria phylum, so-called sub kingdom of bacteria. But this is we found new thermophile of another phylum called Deinococcus thermus, and the name is this one. But the question, of course, why we did not recover Hydrogenophilus thermoluteus, as I told you before, for two reasons. One reason I showed you before, that the eye segments 1 meter, 3707 sample 1 meter. We analyzed one segment, another. Maybe we were not happy to point to the segment of ice which contain this. But another reason, we use it more sophisticated, so-called highly degenerate primers. And maybe this is the region of region is the same, but the primers were different. And maybe this is also the reason. But it's not the matter. Why? Again, no picture. I don't know why. It should be globe. There's globe. No. You see here, we found until now Hydrogenophilus thermoluteus and Thermus species, two species. This Antarctica. This location of our laboratories. It means France, French, Grenoble, Russia. And in blue, this is Meothermus. There is no in red. Hydrogenophilus thermoluteus. Some area overlapping. Both this thermophile, mesothermophile. It means they do not live in 100-degree water, but optimum temperature is around 50-60 degrees, not -2, of course. And the outcome of this study. So this is stuff, Meothermus hypogeus, new thermophile, may or might represent indigenous cell population inhabiting fouls offshore of the Lake Vostok, not the lake of course, namely. And this could provide prospect. Why to not search thermophile there? Because if you would like to search life on Mars, on Europa, on Enceladus, you will be faced to very hard, severe contamination problem. With thermophile, it's easy. It's easy to escape this problem because thermophile is specific. And about the Lake Vostok, should be a kind of thermal life in default offshore of southwestern Lake Vostok. These two candidates. But for the lake, as well as Enceladus, Europa, subsurface ocean, Lake Vostok microbiota and those microbiota, provided the ocean is in contact with a rocky background, can contain some thermophilic bacteria inhabiting deep rocky fault with geothermal activity. So it's a way to study, to try to study extraterrestrial life, focus it on thermophilic life, despite this is icy moons or icy planets. And just regarding the technique, of course, Oxford nanopore technique, third generation technology outperforms Senja, of course. But of course, it's much less accurate, but million reads vis-a-vis 100 maximum or 200. Prone to contamination because many steps still to prepare libraries, gene libraries, and so on. And not yet suitable for searching unknown extraterrestrial life. Why? I just why? Because when you get these reads, 1.5 million reads, only half of these reads, or 80%, could be classified. We have a black box, black matter. Some reads could be not classified against our gene bank, Earth's terrestrial microbiota gene bank. Why? Because even our gene bank does not contain all possible microbes which can be met. So it's problematic if you use this technique just in general sense, not focus it on thermophiles. You will get something which are not met on Earth, but it does not mean that it come from the Mars, it come from Enceladus, and come from Europe. And so it's another way to think how to search for extraterrestrial life. I insist it should be the life specifically adapted to specific conditions, and in our terms, this is thermophilic life. And it's possible to collect material from water vapor samples from Enceladus, Europa, and precisely focus it in terms of this material for thermophile. Thank you very much for your attention.
Thank you, Sergey, for your exciting presentation. Do we have questions?
Yes, we have one question.
Mm-hmm.
Sergey also joined to this congratulation. It looks like real discovery, but what I don't understand, you found past life or present life, because if it's thermophilic bacteria, they get used to live in something like boiling temperature. In ice, will they survive, or is it dead bodies?
I answer the very good question. We analyzed just DNA, and DNA was not damaged. Why? If provided it was damaged, our technique will not work. We need to employ specific materials to restore this DNA. It means this is like extant, not extinct. No, I mean in terms of course, 100 years, 1,000 years, not far extinct, at extant DNA, unperturbated, undamaged. In both cases, I would say. Provided there is some damages of DNA, we can try to repair the DNA. Maybe in this sense, by this way, we can recover extinct life, but we are not interested in this.
I mean something more simple, that not DNA damage is complicated, but they're just frozen because of the small temperature there.
No, they both thermophiles do not grow in the lake water because lake water is -2. They have optimum +50, +60. It means they grow alongside the lake in deep fouls where geothermal activity operates, and they could be flushed out from these fouls and get frozen in the ice. Why? Because the glacier come through, it moves. It glacier. The rate of moving 3 meters per year at the surface. But of course, this is 4 kilometers. So it means a glacier moves, and if something flushed out, it could be trapped in the ice and goes over the lake where the Russians driller get the ice. So it means extent maybe maximum 20,000 years. Maximum, maybe much less, but DNA is not damaged. Except, of course, this ice I just damaged. But perfect material to keep the ice undamaged, DNA. And then why? No oxygen. It's depleted completely oxygen. And there is a water. Oxidation could be, but no oxygen, it could survive a long period, DNA still, but not million years, of course.
Mm-hmm. Thank you. Vladimir, your question, please.
Can you tell something about hot springs on the bottom of Lake Vostok, about this area? And is it planned to explore this hydrothermal area, maybe not with boreholes, but geophysical method, maybe?
Merci, Vladimir. Of course, we can suggest, but the Vostok program is now in a pause. I mean, the depth under the borehole about 800 meters of water layer. 800 meters. The lake is huge. I showed you 250 kilometers. Maybe in some places there is some hot, not hot, but maybe warm geysers. I don't know what is it, but we don't know. We can only imagine. We suggest more simple explanation because the lake is semi-graben, geologically graben structure. It could be some fouls just around the lake, where this stuff could be living, not in the geysers in the smokers. But by the way, the water is enriched with oxygen 18. It means that there is a hydrothermal activity. Interaction with rocks.
Thank you. Sergey, I have a question. Can you estimate some other methods of DNA sequencing? What is the best method to work with such samples? I mean, not only nanopore or Sanger, but also Illumina and some others. Can you say something about that?
Yes, Illumina is a very popular method, also millions of reads, but very short. Illumina mostly read very short segments of DNA, just 250 in average. Using this DNA fragment is not possible to identify, to classify what is it. Nanopore technology can read very long until whole virus DNA is if provided this is DNA virus. So it's very long reads, and now very good practice to combine both techniques. Illumina, which read short reads, and Nanopore long reads. And long reads allow you to combine in a right way and the DNA and restore the genome. I did not tell about metagenomic approach. This is not metagenomic. Why? Because still we face to plenty of contamination. If we can just extract DNA and amplify with nanopore, but after that, I did not show you, we will get maybe 100 species, different species, mostly contamination, and it's not possible to restore genome because it will be plenty of overlapping. And at present, there is only two techniques, PAC-BIO, so-called PAC-BIO, and nanopore, which outperform Illumina, I would say. Two. But PAC-BY still require huge, not a huge, but a big equipment. I just showed you nanopore just require the stick, a minine like this size. You can take it in the field, in Antarctica, in space, I showed you before, and sequence there. Of course, you need to have internet connection because sequencing is done in clouds, in clouds, and classification also performs in clouds. So with no interaction, no internet connection, it's not possible to sequence in the field. If there is you have mobile, so you can use your mobile as modem and run sequencing in Antarctica.
Okay, thank you. We have more questions? Not.
No.
Okay, so the next talk of Alexander Guridov is cancelled, as far as I understand, and in this session we have the last one, Daniil Mironov. But Daniil, can we shift your talk a bit later to the next session, next part of the session? Daniil, are you here?
Yeah, of course, not problem.
Is not a problem. Yeah, that's
Yeah.
very nice.
After coffee break, yeah?
Yeah, coffee, exactly, after coffee break. So
Yeah,
now,
yeah, of course, no problem, no problem.
okay, then coffee break now, yeah, and after that we can continue with the Daniil talk.
At 12?
Yes, would be nice, of course, at 12, but I don't know if people would need more time, so we can maybe postpone for 5 minutes. Yeah.
Okay, 12:05, please be here.
Exactly, yeah.
Hello, dear EC. Do you hear us? EC Liu.
Yes.
Yes, thank you. Okay, EC, so you're connected now to the Rome section, so you will be presenting in extrasolar planets as you're in the program. Please, you should go to your mail and find the Yandex Telemost link. So we have this not in Zoom, but in Yandex Telemost. So please go to mail, find the link. I will send it to you one more time. Okay?
Okay, I received, and there's a link.
Yes, there is a Zoom link, but you have parallel sections.
Telemost.
Yeah, yes, this one.
Okay. So I should type this on my website and join that meeting?
Yes, this one.
Okay. Thank you very much.
Oh, because there is another section in Zoom. So it's astrobiology section.
Okay, I know it. Thank you.
Okay, dear colleagues, please take your seats.
Waiting for the audience.
Dear colleagues, can you see my presentation?
Yes, we see your presentation. Thank you.
Thank you. I can start, or we can.
Well, I see people. They are discussing something in the conference room. But it's I think it's we can start now. Yeah. Mm-hmm.
Okay. Let's start. Dear colleagues, it's good to hear and see you. Sorry, I should stop the video because of that internet connection. First of all, I would like to express my gratitude to the organizers of this conference and my special thanks to the convener of the section, Oleg Kostrubenko. And so you can see the title of my report on the screen. And let's talk about biomining. Slide switched.
Yes.
Yes. Okay, thank you. What about biomining or bioleaching, also known as it is a process of in mining and biohydrometallurgy. It's a natural process of interactions between microbes and minerals that extracts valuable metals from low-grade ore with the help of microorganisms such as bacteria or archaea. Maybe microorganisms and their metabolites have been commercially applied in the leaching of metals from medium and also low-grade sulfide minerals for many years. It's not that the recent results of Chandrayaan-3 show high sulfur content at the South Pole, so it is suggested that sulfide minerals may be common there. And also, the industrial use of biomining is based on the basic natural property of organisms to extract and process certain elements for their life activity. Industrial biomining operations are several kinds, depending on ore type, or its geographical location, the metal content, and the specific minerals present, for example, metal oxides and metal sulfides of different kinds. At the moment, several biomining technologies are being used. These technologies are also based on the use of different species of microorganisms. The industry mostly used some species of ferrooxidants, and also this technology prefer some methods based on the use of fungi, and also actively developed about using fungi. And for our work, we also prefer to use Aspergillus niger because I think it will be more prospective than using microorganisms. And why? We prefer use Aspergillus niger. It is known for its ability to produce large amounts of citric acid and other organic acid, which is widely used in the food, maybe, and beverage industry as a flavor enhanced and preservative. It is also used in the production of pharma technology, maybe cleaning agents and cosmetics, and Aspergillus niger can also produce other organic. Aspergillus niger is a prolific producer of enzymes, including amylases, proteases, and cellulases. These enzymes have various industrial applications, such as in the production of biofuels, also maybe textile food processing. But Aspergillus niger but Aspergillus niger is also used in bioremediation processes due to ability to degrade and detoxify various pollutants, including heavy metals and organic pollutants. And also, you should know that heavy metals also used in industry and in mining. So, overall, the unique characteristic of Aspergillus niger make it a versatile and valuable microorganism in various industries, and also it can be used in biomining. And I should say that some technologies in biomining are testing with these species. Not in huge volume, but it's like testing, and we prefer in our work to use it in future in maybe biomining processes on the moon. And you should know also some plans for lunar program. As you know, now several countries and space agencies have announced their plans for colonize the moon and also develop it, including resource extraction. On the screen you can see the some plans of the Russian space agency Roscosmos to launch space missions to the moon. Most of the world plans include the creation of habitat bases, explorations of the moon interior and its material, study of ice, and also volatile components. And industrial development includes extraction of oxygen, helium isotopes, and so on. And recent years, growing plants also in lunar soil have been actively discussed. Most of interesting is the development of detective technologies and the development of 3D printing on the moon to create dust-free bridgeheads and printing constructions and household items from lunar regolith. As another promising technology, we propose to development of biotechnologies. One of these could be biomining, why not? And our institute, GEHIRAS, has begun work on the development of lunar biomining technology. Our work is in initial state, first step, and includes selection of optimal conditions and methods for analyzing and monitoring metal leaching. So far, we have managed to conduct an experiment with the following conditions to test the effectivity of the proposed technology. Experiments were carried out in the biological leaching of a chemical mineralogical analog of lunar regolith, VILH1. It's our sole analog which used for different experiments. And Aspergillus niger, you can see the name of this species, was used in biological agent. It was chosen due to this high production of organic acids. The synthesis of the following organic acid was demonstrated for studied culture during cultivation on a liquid Chapik medium. And you can see some other parameter of the experiment. Biological leaching was carried out in sterile petri dishes with 10 grams of analog regolith, calcium for 6 hours at 250 by Celsius. One stage leaching, 5 milliliters of spore suspension consisting of Chapek's medium with pH 6.5 and about 10 million per milliliter of Aspergillus niger spores was directly added to petri dishes with analog until 120% of the total moisture capacity was reached. For two-stage leaching, 5 milliliters of the medium filtrate was added to petri dishes at the week of cultivation of the Aspergillus niger strain. The temperature was maintained in a range of 22 or 25 by Celsius. 3 samples for analysis were taken from the general pool and so on, and at 14 days. Additionally, an experiment was carried out on the leaching of metals using solutions of organic acids in concentration similar to those produced by the studied strain of Aspergillus niger. And the next slide, this kind of work is very important to understand the composition of the material. So I should give some information about this analog. You can see many of these characteristics on the slide, but mainly is bulk chemistry. The bulk chemistry of material of Zasnikovskaya deposit. It's the main material of our analog, of our simulant. Is very similar to lunar rocks in many aspects, but the difference in the high alkaline concentration and in you can see in many comparison with some analogs, it is closer to lunar rocks. Also, it is noted that the most analogs of continental lunar rocks have similar alkalinity, and this material is classified as intermediately rated to basaltic trachandezites, andesites of normal and moderated calinity, which is fairly close to lunar rocks. The main minerals you can see on the table used by according to X-ray diffraction analysis. Sorry. I should say about plagia classes it's the main minerals of our NELAC and also in Moon. Plagia class with anatite molecule more than 90% are represented on the Moon, but on the Earth such minerals with such content are less common, more than less common. The composition of our plagia class is 54% of the rock in labradorite, with concentration of anatite molecule mainly 56 to 61%, reaching 72% in some phases. 26% in our NELAC is represented by andesine. The content of anatite molecule is 30 to 50%. It is not that the contained minerals and are characteristic of lunar andesite in general. For our work, we used the following particle model. I should say that the particle size is also important for biomining. I can say that it works like grinding coffee, milling coffee. The smaller the grind of coffee, the richer the flavor of the coffee. Yes, something like that. It is standard for our NELAC, but I know that the model can be modified depending on the goals and tasks. And you can see the grain size distribution modeling demonstrated that the particle size is generally similar to that of lunar regolith based on Apollo 16 data. And let's talk about some results. Comparison of the average values of the t-test XRF analysis of the content of oxides, for example, aluminum, titanium, ferrum, magnesium, magnesium, in our sample did not show statistically significant differences between the control, one-stage and two-stage bioleaching. Measurement of the elemental composition of various minerals using scanning microscopy, you can see on the slide, also showed no statistical differences in the average values of the t-test. It is, we can say that it is really weak leaching in our condition, and way leaching may be due to the short duration of the experiment and incorrectly selected pulp density. To obtain the correct parameters for the operation of the technology, it is supposed to test experiments using different pulp densities and longer time intervals. Also, we could get incorrect results due to indirect analysis. We think that we should not analyze soil, we should analyze water and so on, by the difference in the content of element in the solid part between control and experimental samples. To obtain more correct results, it is necessary to use methods of direct analysis of the results in solutions. Yes, and you can see this scanning microscopy results before its left picture and after its right picture in the experiment. It is not that such elements as titanium, magnesium, aluminum, iron, and so on are particularly not huge and not have huge, except that the current parameters of the experiment. I should say that our results are not very good now. although some experiments with the terrestrial basalts and all under this condition have shown more positive results. And chemical composition of those basalts and our basalts is very similar. But so we're working on improving the technology. At the moment, we are already experimenting with new conditions, and we hope the future testing of this technology will show more positive results. Here you can see our highlights of my presentation. The main goal, the main thing you should know that it is really prospective technology in extracting minerals and metals from the moon surface, from the moon soil. Despite the success of the method, this proved to be incapable for lunar soil. New results need increased suspension volume, increased temperature, and increased amount of time, as we think, and new experiments are already in progress. And some acknowledgements, our team would like to acknowledge the staff of the Laboratory of Meteoritics and Cosmochemistry, Geohiras, for assistance in some analysis. And that's all. Thank you for your attention. If you have a question, I think I can answer some. And here's my contact for maybe another questions or maybe collaborations.
Yeah, thank you, Daniel. Very interesting presentation. So I have a question. Actually, this lunar program is very exciting, and of course, all biologists, microbiologists, astrobiologists can contribute to this program a lot. I think it's very interesting to use different biological activities of microorganisms. You're talking about bioleaching, which is also very important, but I think we can also consider such activities like degradation of different organic compounds, wastes. Maybe also very important and interesting direction is creating a kind of association between fungi and plants in order to increase the productivity of plants, which is important for the team who is going to work in lunar station. Are you going to plan something like this, some other experiments, not only bioleaching?
Yeah, maybe we can work with it. I should say that bioleaching is not used for mining. It is also can be used as, for example, preparing soils for growing plants. Maybe it looks like some mycorrhizal processes and so on. We can work with that. Yeah. And we plan to collaborate with many teams, of course.
Okay, thanks. More questions, please?
We don't have any questions. Let's thank our speaker again.
Okay. So what we have now? We have
a talk, survival of microorganism over 2 years.
Survival. The first talk from Elena Deshova is cancelled, so now we have the second talk, yeah?
Yes, and it will be presented by Vladimir Pichokin.
Okay, please, Vladimir.
Hello, everybody. I'll tell you about cosmic experiment test conducted on the ICC during 10 years. Thank you. The outer surface of the International Space Station is an ideal experimental platform for studying the survival of
Sorry, Vladimir, maybe you can,
in space.
bit closer to the microphone while the sound is not so good now.
So, latest number?
Yeah, it's better,
Okay.
thank you.
Okay. The outer surface of the International Space Station is an ideal experimental platform for studying the survival of microorganisms in space. A lot of experiments carried out in the space vacuum near the ITC study the potential of biological objects to adapt and survive in space conditions. German experiment ExoStack showed if shielded against solar UV spores of Bacillus subtilis were capable of surviving in space for up to 6 years. And 30% of Bacillus subtilis spores survived in the nearly 6 years exposure to outer space when embedded in salt crystals, whereas 80% survived in the presence of glucose, which stabilized the structure of the secular macromolecules, especially during vacuum-induced dehydration. There is a possibility of interplanetary transfer of microorganisms inside meteorites, the so-called hypothesis of lithopanspermia. Russian biologist experiment showed if shielded against space UV is 100%, biological test objects of organisms belonging to various taxonomic groups, from bacteria to vertebrates and plants, were capable of surviving in space vacuum for up to 3 years. But a number of changes in the ultrastructure of both eukaryotic and prokaryotic microorganisms, as well as their biological properties under the influence of open space, have been revealed. European exposed experiments showed that microorganisms in polymer bags placed in a metal 3-layer track can survive under the influence of cosmic UV radiation. In this experiment, biological objects were protected from the effects of the cosmic vacuum. Microbiological spores in artificial meteorites can survive the journey to Mars. Japanese Tanpopo experiment showed that when bacteria of Deinococcus redurans species are laid into aluminum wheels, the bacteria in upper layer does not survive under the influence of UV radiation. Experiment test starting in 2010. Stage 1 was sampling from the outer ITC surface by astronaut surface. During the operation of the ITC, it was discovered that the outer surface of the modules was contaminated with the cold space dust caused by various factors. We have conducted research to determine the physicochemical properties to detect biological structures and viable microorganisms in this cosmic dust. To do this, starting in 1910, the first stage of the test experiment was conducted. In this experiment, the possibility of a regular sampling of the cosmic dust in its natural states from the outer surface of the ICC and delivery of these samples to laboratory for next research. A result of the first stage is the biological studies have shown the presence of DNA of biological structures and viable units of microorganisms isolated from samples delivered to the laboratory. Analysis of the data obtained showed that in these samples, the DNA of archaea, spore bacteria, and fungi, as well as viable units of bacteria of the species Bacillus and fungi Urobasidum pollulans were most often found. The stage 2 consists of preparation macrobiological objects to exposure in the outer space near the ICC. We determined the number of surviving microorganisms deposited on various surfaces during sudden pressure drop. The studies were conducted with spores of various strains of bacteria and fungi. The spores were deposited on the surface of metal plates, on metal rods, on cotton wool wrapped and fixed on the surface of the metal rails. To ensure a sharp pressure drop from 1 bar to 3 times 10 to the power of -7 bar, a vacuum chamber was used. All strains of microorganisms were exposed to different ultraviolet radiation fluences. As a result, the following microorganisms were selected for subsequent tests in outer space with enforced duration of 1 and 2 years. Bacterial spores of the Bacillus subtilis species isolated from the outer surface of the ITC portfolio. Fungal spores of the Oira Basidium pollulant species isolated from the outer surface of the radiator of the ITC temperature control system. Here of the Methanotharcinoma, the S60 species, collection strain from Russian collection of microorganisms, VKM B16360. Cotton wool wrapped and fixed on the surface of metal rods and contaminated with microorganisms was used for exposure. The stage of exposure in outer space carried out from 2017 to 2019. This device installed on the ITC outside, the transport module, 3 rails wrapped in cotton wool contaminated with microorganisms. For transportation of microorganisms and the exposure in outer space, device test was developed. During the spacewalk, the astronaut installed the test device on the outside of the ICC. The installation location device have showed this slide. After exposure is completed, the astronaut placed the test rods with cotton wool in sterile transport modules of device test. Then these transport modules were delivered to the laboratory. Results of our experiments showed on this slide. Within the first year, the number of CFO bacteria, archaea, and fungi decreased by 3 orders of magnitude. During second year, the number of CFO of all microorganisms decreased by another 2 orders of magnitude. We made the same image of methanoid carcinoma cell after 2 years exposure. The same image showed the formation of like methanoid carcinoma cells after 2 years, characterized by presence of a multiplayer thickened cell membrane, which is not absorbed in the control strain. The experiment for irradiation, gamma radiation of strains from after exposure, we have done in the laboratory conditions. This table shows that subcultures of fungi delivered from international ICC surviving the dose of gamma radiation about 5 kilogray and 10 kilogray. So some units can survive 15 kilograms irradiation. There are no, мы также we investigations the effect of stress factors on the fungal strains, such as temperature factor and chemical factor. Growth of spores after 15 minutes of exposure to different temperature, you can see in the table from temperature dependence. We have now revealed difference between control strain, isolated strain, and subculture strain of fungi. The growth retardation zone of the isolated strain and isolated strain subcultures are significantly lower than those of the control strain of VKMF1116 under the action of hydrogen peroxide and 1% chlorine is shown in the table, right table. Conclusion. As a result of the space experiment test, the possibility of survival of bacteria spores of species Bacillus subtilis, fungi spores of special Basidium pallulans, and methanoforming archaea of the species Methanoscarcina mazae S6T, deposited on the cotton wool fixed on metal rods, was proved when exposed for 2 years in open space near the ICC without the use of additional protection barriers. For the first time, the features of surviving archaea Methanoscarcina mazae S6T strain were discovered. After 2 years for exposure, Methanoscarcina cells were found to have its quiche-like forms characterized by the presence of a multi-layered thickened cell membrane. A specific feature of population variability was revealed in 30% of strains of Basidium pallulans species fungi to increase resistance to gamma radiation effect after 2 years of exposure in open space. We showed that the reaction to the effect of different concentrations of hydrogen peroxide and 1% chlorine in the control strain of Basidium pallulans VKMF1116 is more pronounced than in the strain isolated from the outer surface of the ICC and strains after 2 years of exposure in outer space. Thanks for your attention.
Thank you.
Thank you very much. Very interesting presentation. It's amazing actually how we can find microorganisms or spores outside the space station in space. I simply think it would be interesting to do the same experiment with dust on the moon, like on the station located on the moon, to try to find any microorganisms on the dust outside the station, if it's possible or not, kind of checking the panspermia hypothesis. And of course, for me, it's interesting that methanogenic archaea can be so resistant, because methane is one of the important biomarkers, and we're always considering methanogens as the best candidates for colonizing different types of extraterrestrial ecosystems. But well, that was my comment. Do we have questions?
I don't see any questions here.
So maybe I have another question. For me, it's interesting. What kind of experiments are you planning to do at the space station further? What are the plans? Because for me, it's very interesting to see the results about the resistance of different microorganisms in space.
Yes, this experiment is continued now.
Sorry?
This experiment now is continuing.
Yeah, but I mean the same experiments. But maybe you're going to use some other microorganisms, or to choose some other conditions.
I hope that we can modify the experiment.
So they are continuing, and they. So you're going to continue this work with microorganisms in space, right?
I don't understand that. More slide. Not rapid.
Okay. Do we have more questions? Maybe from the
We have
audience.
one question. Lisa,
Zoom
could you please?
or in the conference hall?
Yes, we have one question.
Okay.
Hello.
Perfect.
Hello. My question is. Okay, I have a question. I have a question about anabiotic state. When you culture your microorganism outside the station, they probably enter to anabiotic state or not? No? No any anabiosis? Another question, when you analyze survival rate, yeah, do some cells do not propagate? Maybe they alive but do not propagate. Did you test for DNA? For DNA content?
The bacteria is now in investigations, and we plan to fungi.
Yes, okay, merci.
Okay. Thank you. Thank you.
No questions? No questions,
No
yeah?
questions.
Ola?
Yes. Thanks again.
No questions, yeah?
No questions.
So as far as I understand, that was the last presentation before lunch, because the talk of Ilyin and Podubko is also cancelled.
Yes.
So
The speakers are
we are
very
finishing
sorry.
this session, yeah, 15 minutes before, according to the schedule. And if maybe the organizers have an announcement, any announcement?
No, we should have lunch according to other schedule.
Okay, then we go for lunch, and we'll meet again at
2 p.m.
at 2 p.m. exactly, yeah. Thank you.
Yes, 2 p.m. sharp.
Mhm. Hi, Sochan.
Hi, Oleg. Just let me get a glass of water, please.
Okay,
I might need it.
no problem.
Hi, Sohan.
Hi, Vladimir. Good to see you.
Nice to see you too.
Yeah. Are you busy? How's
A
things?
little bit.
Great. That's the way it should be. When I haven't started writing the report for the microbial world, but I will start this week actually, because I've been very busy with too many things happening. And as you know, that we're trying to get the African project off the ground as well. So it's only today, I've been in negotiation today, that actually we started to make progress. And as soon as I finish here, I got to make some telephone calls, and because I want to contact some suppliers of these instrumentations I need for the African project, yeah?
Okay.
I think we have to start the second part of our session. People are in the room already, yeah?
Okay, I'll share my slides.
So yeah, the first speaker of the second part of our session is Sochan Chita with his talk. So it's about irradiation of methyl cyanide, yeah, with 200 with something. So you can
yeah,
tell us
okay.
yourself. Yeah, please. Yeah.
Yes,
Yeah.
please share your screen.
This is a new computer. Don't tell me. Don't tell me. This is a new computer. I just bought. Cancel. Cancel. Start again. Okay. Entire screen Windows. Yeah. Okay. I don't.
Do you have on this share the screen button? I don't know how it's demonstrate.
Yeah, but on my, oh God, can I ask next speaker to go while I change over computers?
Oh,
huh? Or not? Because they
The second talk is by Oleg, so.
Okay, so then probably I can start with my talk, yeah, and then Sochan.
Sochan, sorry, if you want, you can send your presentation to the chat, and I can share it for you after Oleg's talk. Okay.
No, it's okay. I got two computers. The one that
Okay, no
I'm
problem.
on now, I've just started using it, but I didn't. I've been listening. I've been using. I have been using Zoom, but I never shared anything with it. But I am trying to share the screen, but my PowerPoints aren't showing. They're
Okay.
not showing. Just 1 second. If I see if I can try once more. So this is the one I want to share. And so
Okay, so I think I will start, yeah?
yeah, please start, because I can't do it.
Okay. Okay, dear colleagues, today in my presentation I would like to discuss issues related to the development of astrobiology in Russia. If we are talking about what ideas and what scientific trends preceded the emergence of astrobiology in Russia, that I think are currently influencing it, then first of all, it's such a scientific and philosophical direction as cosmism. Cosmism originated in ancient culture. The term cosmism is of ancient Greek origin. Greek thinkers viewed the cosmos as a complex and ordered entity. In the Middle Ages, Giordano Bruno wrote about multiple worlds in the universe, and later various aspects of cosmism were developed in the works of Humboldt, Fiske, Bergman, and Alexander. At the same time, cosmism was developed in Russia in works of religious philosophers such as Fyodorov, Solovyov, Florensky, Berdyaev. Cosmism was also reflected in the works of artists, painters. Here you see a beautiful painting of Sardan from the Armavela artist group. So ideas of an inextricable and multifaceted relationship between the macro and microcosm, the idea of a space that doesn't know emptiness, biocosmos, in the painting A Symphony of Space, the beautiful picture. And of course, the most important stage in establishing astrobiological research in Russia is the contribution of scientists, cosmists. The fundamental works and concepts which are presented in this table. You see here Tsiolkovsky, Oparin, Vernadsky, and other cosmists with the different concepts. The last one in the table is Gavril Tikhov, who actually introduced the term astrobiology for the first time into scientific practice. New science, astrobiology. You see here the illustration to the paper in the journal Science and Life, where this term was first appeared. Tikhov also published the first book of astrobiology and described for the first time the main tasks and prospects of astrobiology. So I want to cite some of his statements to show the basic significance for the development of astrobiology. So life is an extremely persistent and stubborn phenomenon. It can exist in conditions very different from those on Earth. Life can and should exist not only on the planets of the solar system, but also on the planets that undoubtedly move around the countless stars found in the infinite universe. So actually, this is a description of the issues that modern astrobiology studies. The next citation is the close connection of astrobiology with astronomy, physics, chemistry, and biology will unite the efforts of researchers. Actually, the study of life on Earth and on other planets will merge together. This is astrobiology is really a multidisciplinary field of study. So the third quote I would like to present is about the necessity of creation of a special institute of astrobiology, which would concentrate the efforts of different scientists. It should be very good equipped, and actually will do very interesting, a lot of interesting research, and also all these things are actually implemented in astrobiology in Europe and in United States. You see here a kind of scheme, how it's organized, how astrobiology is functioning in Europe. This scheme shows the structure of European astrobiology, in which ESA, European Space Agency, plays a major role as a sponsor. EANA, European Astrobiology Network Association, is active and organizing annual conferences, and the European Institute of Astrobiology, EAI, has been created already and now operates on the basis of financial support from European universities and has working groups. In US, the space agency, NASA, also plays a major role. As you can see, astrobiology at the present stage needs a stable external sponsor who will be interested in the results of such research. On this slide, you see the structure of the European Institute of Astrobiology. There are two types of working groups: scientific working groups and active working groups. Actually, they cover the entire range of possible areas. In addition to scientific areas, important areas are education, outreach, and industry. These elements are very important for the stable development of any scientific discipline. Here you see an example of complete astrobiology course at the University of Edinburgh. All main topics of astrobiology are presented. In pink, some key lectures are marked that could be actually combined into a shorter course. This full astrobiology program can be used as a basis when developing courses for universities. There is also the practice to create special courses in astrobiology focused on the main direction of different universities. For example, in Germany, courses in astrobiology at various universities and research institutes are integrated into other areas as an addition. You see here, for example, the astrobiology is included in bioorganic chemistry, astronomy, geomicrobiology, planet research, physics, biophysics, and so on. The concept of knowledge triangle. This concept reflects the need for effective interaction between science, education, and business to stabilize the direction or discipline under consideration. It's based on the idea that knowledge created by universities should be in demand in the field of science or industry. In Europe, activities within astrobiology include, of course, educational courses, summer school, laboratories and working groups, some methodologies and devices and space program, and so on. In Russia, there are problems in the area of creating and further stabilizing such an astrobiological triangle. There is no effective education at the university level, almost no connection with practice. Additionally, there is no stable source of external financial support. In Europe and the USA, there are space agencies, first of all, as well as universities and research institutes, which in turn receive funding from scientific grants. In general, the situation with financing astrobiological research in Russia is complex, specific, and requires additional discussion. So far, all work related to astrobiology is being done essentially from grants received in other scientific fields. This slide shows the results of an analysis of the structure of the main directions in astrobiology in Russia according to the percentage of presentations, oral and poster, at Russian conferences that include astrobiology. As you can see here, the main directions are research into various research about various extreme terrestrial ecosystems, research at orbital space stations, and conceptual work on the origin of life and its possible existence in extraterrestrial systems. You see here the percentage of different types of main directions in Russian astrobiology. This slide provides more detailed results in some key areas. As you can see, among extreme ecosystems, the largest number of studies have been done in cold and cryo systems, which correlates well with the geographical and climatic features of the territory of Russia. Accordingly, the main extraterrestrial model system studied by Russian astrobiologists is Mars. This slide shows a list of possible astrobiology working groups, being a result from this analysis I presented above. Such working groups, similar to the European experience, could work more focused within the areas and at the same time interact with each other in Russia. And here there are possible tasks that such working groups could perform. So we can say that scientific astrobiological research is carried out in different Russian institutions, but we can't say the same about education in the field of astrobiology in Russia. The only university where astrobiology is included in the educational program is Dubna State University. It's included in an elective module as a part of the radiobiology program. At Moscow State University, there is a program in space biology and medicine, which deals only with some aspects of astrobiology. I think that's all as far as I know about the education in Russia. Of course, we still have a lot of work to do here. On this map, I present the main extreme ecosystems related to astrobiology in Russia. I think it's necessary to create a detailed catalog of such systems for the further study within the framework of astrobiology, as it is already being done in the West. You see here many interesting ecosystems typical for Russia that can be a subject to study, including, of course, orbital systems. And on this slide, I would like to propose a list of tasks to be done for the further development of astrobiology in Russia, according to what I said before. You see, there are many different items, but in conclusion, I would like to point out the last item. NORCEL is an international association of scientists interested in the origin of life and its distribution in the universe. I'm also a member, you see, of this organization, and can say that we have done quite a lot of work altogether to unite all enthusiasts of the above-mentioned field, created several projects, one of which, Microbial World Network, I am in charge. Another product of our activity is a 3-lecture course on astrobiology, which can be taken as a basis for all universities wishing to provide students with basic knowledge in the field of astrobiology. This course of lectures can be further expanded or focused or adapted to the needs of university. So it's the first module definition, structure, basic directions. The second is extreme terrestrial ecosystems and limits of life. And the third is extraterrestrial systems, searching for life in outer space. So there is a lot of work, and we call or encourage to start doing it together. Thank you for your attention. So please, question.
Any questions here, Niki? Ah, yes. We have one question, actually.
Спасибо. Oleg, many thanks for your presentation. I want to say, maybe not a question, but little comment and observation. I think your idea and this idea at all is extremely relevant these days. But I would also like to point out that the formation of such program must necessarily include geology, geography, and maybe other earth science, because as you know, the formation of life on Earth took place in context of many Earth processes. So in future, it is necessary, I think, to include geological, geochemical, and similar Earth sciences into this field. And as you know, maybe there are many geologists, and maybe NASA, European Space Agency, and other space agencies too, who are involved in astrobiology. And I want to say that in GeoShiraz we also have a little astrobiology group. Many thanks.
Yeah, thank you very much. That's what I told you about before. So let's work together and share ideas. That would be perfect.
Sohan, hi.
So I see the hand from Vladimir. Yeah.
Yes, Oleg. Thank you for integration of the data, a lot of data on astrobiology. What further? What do you intend, how you intend to expand this knowledge, this suggestion further within Russia, I mean?
Because I can share my ideas only writing a paper and talking to people who probably could do something, yeah, because I am not the main person to do something, but if somebody could do something taking into account my my presentation, that would be very, very nice. Yeah, thank you. Sohan.
Yeah, thank you very much. Excellent presentation from what I've seen, because I was communicating between the two computers. The other computer just restarted. It downloaded a lot of stuff. But anyway, my input is this, purely from the NOCEL Network of Researchers on the Chemical Emergence of Life's point of view, is this, that we are actually involved in not necessarily astrobiology exclusively, because the Africans want astroscience. They want astroscience. And so the 3 units that you have written, Oleg, they will be implemented in Africa. And today I just completed, this is another reason why I was running late actually. I've only just completed today discussion on how the apparatus should look like, and we will actually implement these apparatus first to begin with in Nigeria, then Ghana. These 2 countries are on the west coast, and then we go to Nigeria and Zambia on the east coast. And so basically what I'm actually trying to say is that we're on the verge of starting astroscience education in Africa. And it'll be great if our Russian partners could also join in, because I'm going to be truthful and let you have it as it is. I'm not going to, in England sometimes we use this word, sex it up. It means I'm not going to, it means I'm not going to make it nicer just for your ears. I'll tell you as it is, as it comes. The thing is this, with the rest of the world is this, especially the Western world, is that they are too busy, right, to deal with anything to do with Africa. There are some people working in Africa, but they will not be as much as what NOCEL will be, and it will happen, I now suspect, by middle next year, around June of next year. And so I'm actually inviting Russians to join in, although the problem with the rest of the Western world is this. They get excited, they're like kids, you know. They get excited about everything. And when you really push comes to shove, they sort of, whoa, wait a minute, we didn't ask for this. Do you know what I mean? They kind of try to get out. I said, listen, it's free world. You know, you're expected to leave. You don't have to join us. But I invite the Argentinians. I invite the Russians, and of course, rest of Latin America as well, to join us in this endeavor. And so let me just get to my other computer and just put something on. So I think I've done my
Sohan, can you maybe simply start your presentation and say us all together, yeah, because I think
we have
we
one
don't
more
have time.
question from Lev Zelený.
Oleg,
okay.
thank you for good plan for developing astrobiology. And thank you, by the way, for mentioning Russian cosmism. We are working on it. I sent you after the meeting package of our presentation, and very good if you bring the founder of astrobiology, good to write paper about him, because he is unknown and his ideas discussed as strange, so you bring the justice back. And what I want to say is, in Russia we say all initiatives should be punished. Yes, and I think the long road always starts from the first step. And what I suggest you to organize a bi-weekly seminar on astrobiology. We are ready to provide a key platform on this, and this will bring together people from other institutes. Also, we will be very happy to hear the news from our Western colleagues, which we somewhat is isolated now. So if you are ready, it's a good idea, and finally it may be result in after 1 year, maybe 1 year and a half, to really bringing together this community here, and we start to accomplish your program you mentioned today. So if you agree, we could discuss the technical details. Thank you.
Thank you very much. I agree.
That's fantastic, actually. Yes, I'm willing to go ahead. All systems are go. All the engines are ready to lift off. And thank you very much. Sorry, who was that speaking?
You.
No.
You mean before? This is Lev Zelený, actually the head of this conference.
Oh, right. Fantastic. That's great. Look, I'm still having problem with the.
Yeah, Sokhan, you can actually your presentation if you're ready. Yeah.
Yeah. Just
can you
just bear with me 1 second, please. Come on.
Or we can postpone again.
No, I've got connected now. I've got connected with both computers. Join with the.
Fantastic. Something strange is happening. Okay. But can you unmute your microphone, Sokhan?
We see your screen.
Can you unmute yourself, Sokhan? Sokhan?
Yeah.
Yeah. Now it's okay. Yeah, please start. But yeah, tap the button. Yeah, for the full screen. Yeah.
Okay, we're in business.
Yes,
it's okay. Yeah.
It's working. Yeah.
Okay, I'm sorry about everybody. These things happen from time to time. My name is Sohan Jita. I'm the founder and chairman of NOCEL, Network Researchers in the Chemical Emergence of Life. Now, today I'm going to talk about irradiation of methyl cyanide at ice at 15K with 200 electron volt protons. So here we go. And methyl cyanide was found in 1971, and then it's been also found in 83 dark molecular clouds, as well as in comets, and it's of astrobiology importance in the sense that it may have provided some of the chemical necessary for the emergence of life on the Earth. And one of the example of dark molecular cloud is this is the commonest thing that everybody knows about is the Horsehead Nebula. And the reason it's dark here is that because it's made up of interstellar dust particles as well as the 80% hydrogen by mass, 19% helium, and less than 1% other heavier elements. And I've already said something about interstellar dust particle. They are 1 in a million for every 1 interstellar dust particle for million hydrogen in 1 cubic centimeter of space. Pressure in these places is something like 10 to the minus 13 millibars. And usually, a lot of the papers are written from 15K, from 10K to 15K temperatures, 50K temperatures of these places. Of course, the other people, some of the papers go even to 100K as well, 10 to 100K. And so astrochemistry, I'm only going to talk about solid phase chemistry, but in Africa, to digress a little, in Africa, we will be promoting gas phase chemistry. Doing gas phase, this is the laboratory that we're going to set up in Africa. So anyway, interstellar dust particles are made up of 80% silicates, as everybody knows, and the rest being sort of carbonaceous material. And these IDPs, interstellar dust particles, are surrounded by icy mantles. Within the icy mantles, there are organic and inorganic molecules present. I was going to say dissolved because it's not water, it's solid. So therefore, within the icy mantle, there are both inorganic and organic molecules. And solid phase chemistry forms about 20%, 45% of the total gas phase chemistry, solid phase chemistry, astrochemistry, I should say. But anyway, the important thing here is that is the formation of hydrogen, dihydrogen, molecular hydrogen. You need 3 body reactions, and interstellar dust particles are a candidate for the formation of a dihydrogen. And these are the ways the methyl cyanide is formed in space, these dark molecular clouds. So it begins with methyl methanol. Photodissociation takes place. There'll be pair production, which is the methyl, positive methyl ion and hydroxyl ion, negative hydroxyl ion. So the methyl ion reacts with cyanide to form H4C2N plus radical. And then so dissociation recombination occurs, whereby in the presence of electron, you get cyanide made, methyl cyanide made plus elimination of atomic hydrogen. And this is the type of chambers that you would use in order to carry out these experiments. This was carried out in Italy, and we used a 200 electron volt had protons, and what we did was to use a matrix, ice matrix, made from argon. So we sprayed, it's kind of a fancy way of saying sandwich of argon. So we kind of threw some argon at the cold target. And then we sprayed upon this argon, we sprayed the test gas, in this case cyanide, and then we sprayed it with argon again in order to protect any contaminants in gas phase present within the chamber. And so you get this matrix. Okay, that's how we did the experiment, and then we irradiated it with protons. And as you can see in this one, this is just to show you fancifully that astrochemistry begins immediately upon a radiation impinging on the sandwich ice of test gas, in this case being being methyl cyanide. And red one is post-irradiation, black is pre-irradiation, and as you can see, there are a lot more peaks present, which are the red post radiation. That tells you that there is rich tapestry of chemistry taking place. And you know that because the red line in this case here shows the destruction of methyl cyanide, and the black line, which is rising all the time, is methane. Methane is one of the byproducts of, or rather I should say destruction product of methyl cyanide, and it's increasing. And these are the type of molecules that we actually obtained: methane, methylenamine, and catenamine, and cyanoacetylene, hydrogen cyanide, and methyl isocyanide. And you know, I was pondering whilst I was doing this, would you, I don't know, would you consider methyl cyanide to be a new compound? Well, it just rearrangement of methyl cyanide, isocyanide. Would you consider that to be a new product? I don't know. Some would say yes, others would disagree. And similarly, catenamine, if you think about it, this is just a structural rearrangement of methyl cyanide as well. So that would be new product. It's a question mark. Anyway, we compared it with Hodgson and Moore's experiment. Sorry, I got to go back 1 minute. In relation to methylenamine, you know, we're comparing it to Hodgson and Moore, what they observed. And of course, what's in red under Hodgson and Moore, the third column, fourth column in this case, is the red one is from Jericho in 2004, and he observed it, and Hodgson and Moore did not. And of course, we're now also comparing some of the products, the irradiation products, irradiated products of cyanide, and it's also, well, you take your pick. It sort of compares, doesn't it? More or less. The Hodgson and Moore actually used 0.8 million electron volts, whereas we use 0.2 million electron volts, and their temperature was 24 hours 15 K. So I'd say it compares well in relation to hydrogen cyanide, methyl isocyanide, cyanoacetylene, and methane production. Okay, bit of the mechanism for formation of these compounds. I'm not going to read out all of these these reactions here. On the left-hand side, from methane through to catenamine, hydrogen cyanide, and cyanoacetylene. As you can see, that these are reactions. And I should have slapped my own hand, because some of you don't know that I'm actually dyslexic. So every time I see things, what I write to be correct, obviously sometimes they're wrong. And the two box diagram on the right hand side, which is right hand side of mine, so yours should be right hand side too, I think. But anyway, the formation of methyl isocyanide, the formulas are completely wrong because I did nothing. I've only just noticed that, to be quite honest. And more often than not, I don't notice these things. You should see me do crosswords. Sometimes I read a word and struggle for weeks and weeks, only to find out I've never read it properly in the first place. Anyway, so that's how bad it is. And just to remind you, some of these compounds that are chemical reactions I'm showing here, methane, all right, you see the table, here they are again, just to remind you. And the essence of telling you about this, at the 2006 CM minus, that's the infrared band, where cyanoacetylene is observed. And also, so let's see what happens in the next slide. Next slide, if you were to have these cyanoacetylene in the presence of cyanide, OCN minus, this reaction is instantaneous, and actually it sort of forms cytosine, one of the nucleobases necessary in the emergence of life on planet Earth. And this particular reaction was taken for Spiro. I have not put it down. I apologize for that. Anybody want references, please write to me and I'll send you the references. And the other one is the bottom one is one oro reaction, which he did, I think, 1966. And all he did was took cyanide and ammonia and heated it together for about a week or so. And lo and behold, he was able to make adenine. This is the other nuclear basis that is used in biology on Earth. And to date now, presently, we now know that all 5 nuclear bases were made in space. The reason I know that, because I read the latest papers that say so. They've been discovered. So the importance of astrochemistry is really, really important. And I'm sure my friend Amina, she will do a better job than I have, because I enjoyed her talk last time. I hope she's giving the same thing. I don't mind listening again. So the conclusion here is that the experiment shows that solid phase chemistry in space is a realistic possibility. Thank you very much. I'll take questions if anybody's going.
Thank you, Sochan. Yeah. Do we have questions?
Questions? There is one question.
Yeah.
Thank you for your nice presentations. One question. This molecular methyl cyanide is a very important atmospheric component in the atmosphere of Titan. And my question is, how many vibrational modes had this molecule, and how many infrared radiations could be effectively radiate this molecule in the atmosphere, including not only main vibrational mode, but also satellites, 2 and 3 modes together.
I'm so sorry. Can you simplify the question a little, because it was rather long. sorry about that.
Could this molecule consists of 6 atoms? Therefore, there are about 6, maybe more vibrational modes. Therefore, there are the possibilities of radiation.
I know where your question is going. Thank you very much. More than what I reported here, to be quite honest. It's not just me. There are a large number of people done these using cyanide. They've used different temperatures, different types of radiation. For example, there's a few scientists in America. They've used UV light on the same. So they're not that many. But the only other things which I haven't reported, to be quite honest, would have been something like hydroxymethyl, which are made in other. Actually, you can't make hydroxymethyl. Just realize because it contains oxygen. Sorry, it's not more than what I've reported here. Yeah, I know where you're coming from.
Okay,
More
thank
questions?
you. Yes, one question.
One more question. Sohan, you know about this material returned from the Ryugu asteroid, and it's very complicated. Molecules were found, something like acids, and something like nicotine acid and so on, and all this chemistry was in somewhere in deep in this asteroid under surface probe taken, and what are the main energy in your opinion, energetic factors to resulting in such very complicated astrochemical production of practically very complex molecules in asteroids? It's a galactic cosmic rays or something like, and or it's something pre-solar material. What is your opinion?
They're certainly not. It's certainly not UV light, because UV light never penetrates other than on the surface of any dark molecular cloud. But it'll be the high energy electrons, protons, and hydrogen atoms. These are the only ones generally people done the experiments with. For example, we've done experiment with the 5,000 electron volt experiments, and that works wonders. So you don't really need a really high energy. In any case, you can actually work out what's the maximum amount of energy that needed to break the bonds between in these molecules, you know, and so you can work that out theoretically. And the only, as I said, the only one would be operative are the high energy particles. I know that there's a scientist in Brazil. He showed high energy particles such as carbon and others even higher than that, like nitrogen, speeded up nitrogen, that could do the chemistry as well, you know. But I don't believe that, because energy required to speed them up has to be phenomenal. And also, as you know, that these structures are really, really ginormous, and you have to go into the heart of wherever wherever it could, where the planetary systems are being made, where it's needed. I think it'd probably be stopped, but I think sometimes people do experiment for their ego rather than what's practically needed. You know, practical needed is that if electrons and protons can do the job, why not? Why make life difficult? That's what I say.
Yeah, thank you, Sohan.
Okay.
Do we have more questions?
I believe we.
If not, then we can
on.
go to the third speaker.
Just one thing. Richard, good to see you. Sohan.
Okay.
Yeah.
Please.
Good to see you.
Yeah. So Jimena Abrivaya is speaking about astrophysical sources of radiation and habitability in the universe.
We see a screen. Can you
see the screen?
Yes. Can you put it on full screen?
Yes, of course. Thank you. So, thank you very much. First of all, I want to thank to the organizers for inviting me to give this talk, especially to Oleg. And in this talk, I'm going to make a little review about astrophysical sources of radiation and habitability in the universe. In astrobiology, one of the goals is to characterize habitable places in the universe, which is meaning regions of the galaxy or planetary bodies or planetary systems. And these regions are allowing us to define habitable zones, following the requirements of life as we know it. Regarding that, we can define galactic habitable zones or planetary or also called circumstellar habitable zones. Regarding the galactic habitable zone, we can say that it's a region inside the galaxy which is having conditions which are compatible with the origin, development, and long-term existence of life as we know it. And we can define, in fact, galactic habitable zone, which according to some studies, for instance, this one from Weber, 2004, is going to 7 to 9 kiloparsecs from the galactic center. And I'm having some problems with my presentation. Okay, nice. We can also define planetary habitable zones, and in this case, this is more restricted to what we call the liquid water habitable zone. This is meaning a region around the star where we are having the conditions to have liquid water on the surface of the planet. And this will also depend on the kind of star we are having. In astrobiology, we consider that the stars which are relevant from the point of view of life are some stars which are belonging to the main sequence stars, and in particular those which are called FG, K, and M, because the connection we are making is related to the lifetime of the stars in connection to the life that that life is needing to develop according to the reference we are having on Earth. And recently also A stars were also considered for astrobiological studies. And in particular, among these stars, apart from G stars, which are stars like the Sun, which are also very relevant because they are because of the connection we are making with life on Earth and what we can find in other planets. DM stars in particular are relevant for these studies because they are the most abundant in the galaxy, 8 times more abundant than G stars. It is relatively easier to find terrestrial planets around these stars. They are also particularly active, so it's relevant to study the stellar activity. And also the habitable zones are closer to the stars because these are cooler stars than the Sun. And therefore the impact of some conditions like stellar radiation would be more important in planets which are orbiting these stars. And if we watch the set of conditions for habitability, we are looking for the conditions for life as we know it. This is meaning that we need to have a rocky planetary body, a solid surface, also a proper star, as I was explaining, liquid water, organic molecules, energy sources, and environmental conditions in proper ranges. But also one of the fundamental factors we need to study is radiation, because it can have impact at multiple levels. It can have an impact at the chemical level, as Sohan was showing before. We can have radiation which is having impact in the production of some molecules which are relevant for life. And also radiation will define the environment, for instance, the atmosphere of the planet. And at the biological level, we are having that this impact evolve among others. We have in the universe different astrophysical sources of radiation, which are coming from emissions in the whole electromagnetic spectrum and also particles. We are having emissions from stellar events, different kind of emissions from these events, also emissions from accretion processes and also from other kind of processes. I'm not going to talk specifically about each one because I don't have enough time, but I just wanted to mention these ones in color are the ones which were considered from the point of view of astrobiology, as I'm going to show in this talk. And regarding this radiation from astrophysical events, in astrobiology, the studies were focused on gamma rays, on X-rays, and UV radiation. This is in the electromagnetic radiation spectrum, but also in cosmic rays and stellar wind, which is particle radiation. The biological effects of electromagnetic radiation in general are well known since a long time. However, in the astrophysical context, it's necessary to perform specific studies because we have seen with our group that some energetic events can bring different results because of the higher doses or fluencies that these processes are involving. And also, we know in general the biological effects of energetic particles that were in general studied, except for muons, which are elementary particles similar to electrons, but with a greater mass. And the problem with muons is that are difficult to produce because they require very specific sources to be produced, and most of the artificial sources are not able to produce these particles, so they were not really studied. And it is assumed that possibly they will have similar effects to electrons, but this biological impact could be more severe because of greater penetration of these particles. And also we can think in radiation effects, like direct effects on life or indirect effects on life. Direct effects are those at the cellular level, for instance, in microorganisms, or in molecules, for instance, for polymerization of prebiotic compounds. And indirect effects on life, those at the level of the environment, for instance, the atmosphere. These effects can be positive, negative, or negligible. This will depend on the kind of radiation we are having, the amounts of radiation, and the capability to repair or to cope with the damage in the case of organisms. And some examples about direct effects on life, well, negative ones will be related to damage or lethal effects of radiation, damage that cannot be repaired. And a good example is UV radiation. We have different wavelengths of UV. In biology, we classify in 3 different ranges: UVA, UVB, UVC. UVB and UVC are the most damaging because, as this plot is showing on the right side, the absorption of proteins or DNA is at these wavelengths, so they can have impact at this in these molecules. And I'm not going to talk specifically about all the mechanisms of UV damage, but we can have different kind of damages, direct damage or indirect damage, and mostly UVC and UVB are responsible for, well, UVC in general is responsible for direct damage, but UVB can produce indirect or direct damage, so these are very harmful wavelengths for life. And sorry, I forgot to mention the positive effects. We can also have positive effects if we think in evolution. For instance, Sagan proposed in 1973 that UV radiation can be important for evolution because it can induce mutations, and at the same time it can work as a selection pressure. And some other direct effects on life we are having at the level of prebiotic chemistry. Negative effects are related to the destruction of organic molecules, but positive related to the formation of prebiotic organic molecules. And we are having here some examples with interstellar ices and also on interplanetary medium, but also we can have the production of some of these molecules at the planetary surface. And about indirect effects on life, we are also having an example here on Earth, which is related to ozone. Depending on the wavelength of UV, we can have the production of ozone or the destruction of ozone, which is meaning that we can have positive or negative effects. This is part of what they are called the Chapman reactions. The potential also of radiation to reach life on the planet will depend on the possibility of shielding to radiation. We can have shielding to radiation by magnetic field if the planet is having a magnetic field as we are having on Earth. This will shield the mostly particle radiation. Atmosphere mostly will shield X-rays, or depending on what kind of atmosphere, also UVC or part of UVB. But we are having other environmental shields, water in the case of oceans, ice, salt, or we can have also terrain habitats or rocks, which also will shield radiation. And well, also the magnetic field can shield the surface from the planet from charged particles, and this will depend on the strength of the field and the rigidity of the particles. And also the atmosphere in some cases can protect both from particle and electromagnetic radiation. This will depend on the energy of the radiation and thickness of the atmosphere. And also, as I was mentioning, ice and rock can shield against both high energy photons and charged particles. Also, we can have other kind of shielding, which is the biological one, and also in case of organisms, we are having repair mechanisms. And just to mention some of them, pigments can work as shielding, also as radical scavengers. Then we have repair mechanisms, in some cases improved repair mechanisms, as in the microorganisms, the Deinococcus radiodurans, or redundant repair mechanisms. In some cases, we can have intracellular ions, which are also protective for radiation. We can have intracellular salts that also can protect from radiation in some cases, and also we can have the fact that microorganisms can form structures like clamping and biofilms, which will create shielding. And also we can have other features in microorganisms as polyploidy and genome composition that can influence in the capability of the microorganism to survive to an exposure to radiation. So in general, the overall biological effects of UV radiation, for instance, will depend on the balance between photoprotection, damage, and DNA repair. And how we estimate the impact of astrophysical events on life? Well, we can have different kind of approaches. We can have theoretical ones where we use experimental data from literature, for instance, and we make from that mathematical approximations, computer simulations. But we can have also experimental approaches where we use terrestrial microorganisms or molecules, and we expose them to conditions we are not having on Earth. And from this point of view, we have two different possibilities. We can have experiments in real environmental conditions to simulate different scenarios. Typically, these are the experiments that are done in the low Earth orbit. Or we can make experiments in the lab in simulated environmental conditions. I'm going to talk a little bit about these ones, these experimental approaches. Well, we had talk this morning about simulation experiments in space. This is just to mention some of them. Expose-E, Expose-R, also the ESA Stone and Biopan experiments, but there are so many others. In general, what we do is to expose microorganisms of molecules to the conditions of the low Earth orbit, and the samples are returning to Earth for analysis. And we have some advantages of these kind of approaches, like, for instance, we can make exposure to multi-wavelengths or several kinds of radiation in parallel or in combination with other conditions, like multi-parametric simulations, which are very advantageous. But we can have also disadvantages from these approaches, like few control of the doses we are having, because we cannot control sources of radiation. Also, they are more, it's more difficult to have access to these experiments, and they are much more expensive, of course. And then on the other side, we have the simulation experiments in the lab, where we are using simulation chambers, irradiation facilities, or devices. Few of these devices are commercial, but most are lab-made specifically for these experiments. And here we can control not only radiation, but also temperature, atmospheric composition, vacuum. And also we can have the possibility to use facility as particle accelerators. And also in the case of UV, for instance, different kind of UV simulators. And here we have, again, advantages and disadvantages. The advantages is that we can really control very well the experimental conditions, are less expensive and more easy to have access, more easy to repeat the experiments. Disadvantages is not possible always to make multi-wavelength exposures for several kinds of radiation at the same time, or in combination with multiple conditions, as we are having in space. Now I wanted to talk a little bit about the impact of radiation at the galactic level. In this case, we have the fact that radiation can constrain the galactic habitable zone of the Milky Way. And this is because we are having a massive black hole at the center of our galaxy, which is inactive currently, but periodically, when matter falls inside the black hole, is releasing very energetic photons and charged particles. And also we have in this region remnants of supernova activity, and the presence then of particles and gamma ray emissions are limiting the possibility that we are having life at the center of the galaxy. And also from the point of view of planets, we have different kind of environments which are interested to be studied. We have the primitive Earth, where we don't have, for instance, ozone layers, or more UV radiation in different wavelengths than the present Earth. We're able to reach the surface of the planet. We have planetary bodies of the solar system, where we are not having almost atmosphere and no magnetic field. This is also relevant to study from the point of view of radiation. Also, we are having now exoplanets to study where we are having variable conditions, which can include partial or no environmental shielding. And even when this is not about habitability or in the habitability context, but it's also relevant to study from the point of view of radiation is lithopanspermia, especially the interplanetary step. And up to now, the effects on life, which were studied in the astrophysical context, are related to UV stellar emissions. For instance, in the case of flares, there are experimental and theoretical approaches. From the point of view of the stellar wind or cosmic rays, we are also having experimental and theoretical approaches to study the biological impact. And also in the case of supernova and production of gamma ray bursts, we are having theoretical modeling. And this is, I'm not going to talk about this case because this is more theoretical modeling, but some studies have suggested that these supernova events could have had a big impact on early Earth because it could have been responsible for mass extinctions. And also, now as we are having an exponential discovery of exoplanets, some studies are shifted to exoplanets, especially because we are able to detect potential rocky exoplanets. So it's interesting to study habitability in this context.
1 more minute, please, Jimena.
Yes, I will finish soon.
Okay.
So then we are having, as I mentioned before, direct and indirect effects on life, in particular in the case of microorganisms. We have the fact that the biological impact haven't been studied from the point of view of stellar UV emissions. And flares and super flares, which are high energy emissions from stars, are interesting to be studied in this context. And in the case of our group, we are focused in different projects trying to unveil the effects of these stellar flares on life. And we are using interdisciplinary approaches where we are combining biology, astrophysics, and in some cases geology to expand and improve previous studies. And well, we have published one paper regarding to the first experiments to the survival of stellar flares in this journal. I invite you to read it. And also in the context of lithopanspermia, we were studying the protective effects of highlight in the context of the young sun and considering super flare case. And well, thank you very much, and I'm open to questions.
Thank you very much, Jimena, for a very interesting presentation. We have time for 1-2 short questions.
We don't have questions here.
Don't have questions.
No, we have one.
We have one. Okay.
Thank you. Very interesting talk, but you make very strong point on influence of radiation, positive and negative, but we know examples when the life is emerging without any influence of radiation, like black smokers or some life emerging inside the planetary body. What do you think about it? Is it radiation necessary for life?
Well, yes, this I didn't consider non-astrophysical sources of radiation like you are mentioning, because I wanted to limit the talk only for that. But of course, these other sources of radiation are also relevant to be studied. As you mentioned, these black smokers are close to these black smokers are sources of radiation. So yes, I consider that this is very important to be studied as well, and it's important to study this in the context of the origin of life, of course.
Thank you. More questions?
No.
No. Thank you very much, Jimena, for your presentation. Now we go to the next speaker. It's Richard Hoover. Richard?
Yes.
Hi.
Hello, Richard.
Hello.
Yeah.
Let's see here. Am I sharing my screen okay?
Yes, it's
Yeah.
fine.
Okay.
Could you just start the slideshow?
Thank you so
Yes.
much.
Perfect. Thank you very much.
You can see okay?
Yes.
Right. I'm delighted to be able to be with you today by Zoom, and regret that I cannot be there in person, and hope that I will be able to join you again perhaps next year. Title of my talk is Life in the Universe, Extraterrestrial Water, Cyanobacteria, and Diatoms. When, where, and how life originated are the greatest unanswered questions of science. Water is the most abundant compound in the universe and comprises 70% of the mass of all living cells. Water and complex biomolecules in comets, moons and exoplanets, and in fossilized, I'm trying to remove this, that's better. Fossilized diatoms and cyanobacteria and meteorites indicate that life is not restricted to the planet Earth. The complexity of DNA and proteins invalidate the hypothesis that Earth life originated on Earth. It is fascinating to recognize that simple cells just simply do not exist, and life on Earth has been around for at least 3.8 to 4.2 billion years, as evidenced by filamentous fossils that have been found in ancient rocks in Canada. Sharoff did a timeline by linear regression of genetic complexity, and his data indicates that the origin of the prokaryotes occurred at least 3.5 to 4 billion years ago, and the problem is the origin of our solar system occurred somewhere around 4.5 billion years ago. If you do an extrapolation of this timeline back, it suggests that life probably originated around 9 to 10 billion years ago. What is life is very interesting because we know that all known forms of life use energy, DNA, RNA controlled metabolic and enzymatic processes to convert some 20 biogenic elements into complex organic biomolecules that are never found in non-biological systems. The DNA and RNA is comprised of nitrogenous nucleobases with a sugar phosphate backbone, and of course proteins and the enzymes are composed of 20 chiral proteinogenic L-amino acids. The most abundant compound in the universe is water, which you have 2 or more elements. It comprises 60 to 70% of the mass of all living cells, and cells can only metabolize in liquid water. The fascinating point is that life can exist in ice because many of these organisms are able to utilize antifreeze proteins to convert frozen water into liquid water at temperatures significantly below 0 C. Water has this polar bent geometry. The HOH bond angle is 104.5 degrees. It expands on freezing, and that is profoundly important because that means that oceans, lakes, and rivers freeze from the top down rather than the bottom up, and therefore beneath these icy covers of exoplanets and exomoons, there can and are found to be liquid water oceans. The maximum density of water is around +4 C for fresh water and -2 C for seawater, which means that at the bottom of these deep oceans and lakes, the temperature is always the same, 0 C plus or minus 4. All deep oceans and lakes on every planet in every solar system in the universe would be essentially alike, dark, high pressure, and temperature near 0 C, which of course is perfectly suitable temperature and parameters for life itself. The Dawn spacecraft discovered water and organics on the asteroids Ceres and Vesta. Ceres is a rocky core with an icy mantle with of the order of 200 million cubic kilometers of water. So a GRC asteroid similar to CI1 carbonaceous meteorites like Ale, Ivuna, and Orgueil. The 4 Vesta Cornelia crater with gullies and RSL show liquid water flow similar to HED meteorites. The New Horizons discovered evidence for water, oceans, and organics on Pluto and Charon. There are these new mountains, polygons, and ice flows on Pluto which indicate oceans. The red and orange colors have been interpreted as being tolons, but in fact, I suggest that they may in fact be pigments associated with aquatic microorganisms. Sub-ice oceans and cryovolcanism appear to exist on Charon. Here we see a representation of the different kinds of planetary systems with planets with subcrustal oceans. One of the most fascinating things is that the subcrustal oceans of Europa and Enceladus have been attributed to tidal forces associated with the giant planet nearby, causing heat to be produced interior to the ice as a result of tidal forces. The problem with that there is no doubt that works, but there is a problem, and that is there is no gas giant near Pluto. Therefore, the fundamental question is, how is it that Pluto has an enormous subcrustal ocean? Well, I am suggesting that the subcrustal ocean of Pluto probably occurs as a result of heat-producing elements like uranium-238, 235, which has the 238 has a half-life of 4.5 billion years, potassium-40 with a half-life of 1.2 billion years, and thorium-232 with a half-life of 14.2 billion years. Those radiogenic elements are responsible for the heating of the Earth's crust. And in fact, if they exist in the rocky cores of these planets, they could give rise to liquid water oceans existing for time periods similar to the age of our solar system or greater. On the right, you're seeing ice worms, which are coming out of a glacier that I visited in Canada. This is the Whistler Glacier, the Pemberton Glacier, and I did studies of microorganisms inside the ice of the Pemberton Glacier. And every time I took out a core of ice and melted it under the microscope, it was teeming with bacteria. In 1954, the late Sir Fred Hoyle predicted a 7.65 MeV resonant state of carbon-12, which formed by the triple alpha process in superhot helium-burning red giants and CH stars. The fact is that this life-critical carbon was unstable until you got this resonant state in which the alpha particles are at the apices of an equilateral triangle rather than like on the arms of a bent elbow. That occurs about every 1 out of 2,500 reactions, and that prediction has been called the most outrageous prediction in the history of science, and it's also referred to as the Hoyle state. Life-critical carbon, oxygen, nitrogen, hydrogen, and are ejected into space by supernovae and comets that form interstellar dust. These cone particles are very important, and it is intriguing that 99% of all cells can be represented fairly accurately by the chemical formula C4H7O2N1 PO.1 SO.1. So carbon, hydrogen, oxygen, nitrogen, phosphorus, and sulfur are essentially the stuff of life. Phosphorus is profoundly important for life as well. In fact, some have argued that we should think of life as being phosphorus-based rather than carbon-based. Phosphorus is essential for ATP, phospholipids and the phosphate backbone of DNA and RNA. It's formed in phosphorus-rich stars, and PN and PO were detected by ALMA in AFGL 5142 and by Rossini in Comet 67P Churyumov-Gerasimenko. Cassini's cosmic dust analyzer recently detected organics and phosphates. Actually, it was reported recently that they had detected organics and phosphates in icy plumes of Enceladus. These icy plumes are ejected, as we see in the lower left, and they are coming out basically as sodium phosphates. In 1986, I worked with the late Sir Fred Hoyle, and we did a study of the IR spectra of Trapezium Nebula and the galactic center source GCIRS-7. These are shown on the right, and the observed infrared flux is shown in diatoms, in dots from the Trapezium Nebula, and the solid line is the mixed diatom model. This is shown also. You see all of these nice little bumps and wiggles in the lower curve between about 2.7 and 12 microns for the galactic center GCIRS-7 source. In this paper, we pointed out that these results support the concept of a cosmic microbiological model in which diatoms or other similar organisms that produce these silica biopolymers might exist on comets, Europa, and in interstellar space. Energy can be obtained in a wide variety of forms by photosynthesis, by consuming of foods in organotrophic organisms, by consuming of inorganic elements, as in chemotrophic organisms, and the intriguing thing is that the hydrolysis of adenine triphosphate to ADP provides the energy necessary to drive the protein-based molecular motors. Molecular motors play an absolutely critical role in every phenomena of every living organism on Earth. Recent discoveries revealed that there were cyanobacteria in deep dark rocks and that they were hydrogenotrophic. These cyanobacteria discovered in Rio Tinto use hydrogenase enzymes and live on hydrogen, whereas we had always thought of cyanobacteria as being photosynthetic and operating only on photosynthesis using light. Also, we had always thought of diatoms as being photosynthetic, although in this paper in 1986, we pointed out that in Antarctica, diatoms alter from photosynthesis to organotrophic and chemotrophic when they're in the deep night of the polar night in Antarctica and the Arctic. The intriguing thing is that while in this mode, they can consume amino acids directly, and one of the amino acids that they've been found to be able to metabolize is alpha amino isobutyric acid. AIB is a common amino acid in the carbonaceous chondrites. So that introduces an interesting question as to how did they evolve this capability. Life is characterized by very complex molecular machines. Here we see a representation of the microtubules in which these dynein and kinesin is being used to transport information and as a signaling system. Here you see the dynein molecules, which actually have these long stems and foot-type structures where they walk along the microtubules. There goes a kinesin. And so this is the incredibly complex mechanism by which the signal system occurs and the synthesis of DNA, RNA, proteins, biomolecules, organelles, and cells occurs. This is from Drew Berry's representation. One of the aspects of life that is tremendously important is locomotion, motility. Here we see the motility of the vorticellid. In the upper left, there is also cyanobacteria with escaped Marconium replication. In the lower right, you see the swimming motion of Spiracetum americana, which we discovered in Mountain Lake and described in 2003. Due to the viscous nature of water and the Rayleigh number, which for microorganisms similar to these kinds of bacteria is of the order of 10 to the minus fifth, that means that the only way in which they have a swimming in this liquid is by using motors to control helical coil structures like these spirocheta or the helical flagella of bacteria. DNA information is encoded in this magnificent double helix by 4 nucleobases. Adenine is paired with thymine, and guanine is paired with cytosine, and they're attached to this deoxyribose sugar phosphate backbone. The human genome contains of the order of 3 billion base pairs, diatoms of the order of 30 million base pairs, and cyanobacteria of the order of 3.5 million base pairs. One of the intriguing things is that life is absolutely unbelievably complex. This is orotidine 5' phosphate decarboxylase, which is used in the OMP to UMP reaction for the biosynthesis of the pyrimidine bases, uridine, cytidine, and thymidine. In water, without this enzyme, the reaction would occur to convert about 1/2 of the reactants into products, would require of the order of 78 million years. With OMP, the reaction occurs in 17 milliseconds. You see on the right the importance of the sugar phosphate backbone and the Watson-Crick canonical base pairing of adenine with thymine and guanine with cytosine. Of course, in the RNA, the thymine is replaced by uracil, but in RNA, guanine is still paired with cytosine, which means basically that in every cell there is always exactly the same amount of guanine and cytosine, and this is profoundly important for our studies of nucleobases in meteorites. There are extraterrestrial nucleobases, but there are dramatic differences in the content of guanine and cytosine. Very recently, it was reported that on exoplanet K2-18b, the James Webb Telescope had detected evidence of dimethyl sulfide, which is produced only by the cleavage of dimethyl sulfonopropionate metabolite, which is a metabolite of marine diatoms, cyanobacteria, and a wide variety of other phytoplankton and aquatic marine organisms. Dimethyl sulfide and dimethyl sulfopropionate is only produced in biological systems. It's a very complex reaction, and it is not found in abiotic chemicals. This provides very intriguing evidence for the existence of life on exoplanet K2-18b. The James Webb Telescope also used the Einstein ring phenomena to detect polycyclic aromatic hydrocarbons in interstellar dust of a very distant, of the order of 12 billion light year distant, young galaxy SPT 041847. As I said, there have been nucleobases found in the carbonaceous meteorite. This is Martins et al.'s results of Del 13C analysis of uracil, xanthine, and uracil and xanthine in the Murchison meteorite, giving plus 44.5 part per mil uracil and plus 37.7 part per mil of xanthine. This is absolute proof that the nucleobases found in the Murchison meteorite are indigenous and extraterrestrial. Cytosine decays to uracil with a half-life of 17,000 years, and thymine decays to xanthine with a half-life of 1.3 million years. Well, the absence of thymine and cytosine in the carbonaceous meteorites is clear and convincing proof that these meteorites are not contaminated with modern bacteria, with modern cyanobacteria or modern diatoms, because if they were, they would have equal amounts of cytosine and guanine. They would also have all 20 of the proteinogenic amino acids, and only 8 to 12 of the proteinogenic amino acids are missing. Typically, 8 or more of the amino acids are absolutely absent, cannot be detected by the most sensitive mechanisms, and that is again another proof that these meteorites are not contaminated by modern terrestrial biology. In 2018, Irakli Simonia and Dale Cruikshank published a fascinating article, Organic Molecules in the Icy Bodies of the Planetary System. One of the molecules that what they did was they basically looked at unidentified spectral lines in the cometary spectra, and they found that in this, for example, on the left is shown Comet de Vico, and here you see a whole array of wavelengths where peaks occur, and even though they were unable to, it had been previously unable to identify these with minerals or simple Miller-Urey type chemicals, when they looked at more complex chemicals, such as the luciferase enzyme, which uses luciferin, ATP, and oxygen to produce photons, and is what is responsible for the light emanating from fireflies, deep sea bacteria, deep sea worms, and fish. This luciferase molecule occurs over and over again. They also detected evidence of chlorophyll and green fluorescent protein spectral lines in several comets that were analyzed. The problem is that there are 7 genes that code for 547 amino acids that span 1,976 base pairs of genomic DNA of fireflies and bioluminescent bacteria. The probability of linking amino acids together, taking one at random from each of the 20 to get this luciferase molecule, would be 1 divided by 20 to the 547th power, which is another way of writing 0. In 1992, Professor Gold predicted a deep hot biosphere. We now know that there is, in fact, a deep hot biosphere, which extends to over 10 kilometers beneath the Earth's surface, and it is produced by water that has been released by heat-producing elements and by energy released by serpentinization at water-rock interfaces. Over 90% of the biomass of the planet exists in archaea and bacteria, and the vast majority of this archaea and bacteria is present in crustal rocks, which means even though we have long thought that the only way you could find extraterrestrial life would be to find planets, rocky planets with liquid water surface oceans, I submit that is not correct, because the presence of life in deep, hot crustal rocks mean that life can be present on the moon, on the planet Mars, and inside of the crusts of virtually every moon and planet in our solar system, and perhaps even spread throughout the universe. Chlorite serpentine reactions to form serpentinite reactions to form serpentine, which is one of the dominant minerals of the Organe meteorite, produce heat as well, and these reactions show the mechanism for heat production. Energy, water, and hydrogen is released by the long-lived radiogenic heat-producing elements, and this supports a vast ancient biosphere in the deep crustal rocks. Recent discoveries of the ice cube detector in Antarctica show that heat-producing elements are responsible for the Earth being, in fact, an anti-neutrino star, in that there are more anti-neutrinos emanating from the planet Earth than it is receiving from galactic sources. Oceans, atmospheres, and sunlight are not required for life. So the biological systems can extend far beyond what have previously been considered habitable environments.
Richard, one more minute, please.
Okay, there are living cyanobacteria found in the carbonaceous meteorites. This shows the colors of the Minoas Linnea region on Europa, which are consistent with microorganisms. In the deep oceans, there are stalked bathycrinoids that live at great depths. Kerogen is the dominant form of carbon in the Orgay meteorite, and this is typically of the order of Miocene to even more ancient ages. Go through. In 1997, Professor Rosenoff and I started working on microfossils in meteorites. This shows the filament in the Murchison meteorite, and the carbon-nitrogen ratio is in fact consistent with kerogen, with coal. Here we see a representation of the energy dispersive spectra of carbon, oxygen, and carbon-oxygen ratios in the filament of cyanobacteria in the Orgay meteorite. And when we plot that, the carbon-oxygen ratio, it is very similar to bituminous coal. These are calothrix found in the Orgay meteorite and diatoms found in the Orgay Ci1 carbonaceous meteorite found embedded in the rock matrix. They're exotic microorganisms that we cannot identify. This is one which has an incredibly high concentration of fluorine and chlorine. Phosphorus can also be seen in this 2D elemental map. The asteroid Bennu has recently been found to contain organics and water. The same is true of Ryugu, which it was reported contains the evidence for polycyclic aromatic hydrocarbons and dimethyl sulfide on Ryugu. So there is indeed a possibility of transplanetary contamination, not only from within our own solar system, but from interstellar sources, but also there is conceivably a way in which life could exist inside of oceans of rogue planets that have been ejected from another planetary system and travel to the planet Earth, even over time periods of the order of billions of years, with the life continuing to replicate on the interior of the ocean. Therefore, there is a potential for transfer from one galaxy to another by the fact that the living organisms can replicate and grow on the interior oceans protected by ice and thick layers of seawater. Thank you very much for your attention.
Thank you very much, Richard, for your very interesting talk.
Thank you.
Unfortunately, we are quite limited in time, so I would like to ask the audience to formulate the questions very briefly, if they have questions. Maybe we have from the audience, yeah?
No, I don't see any hands.
Mine, Sohan.
Oh, yeah, Sohan, yeah, please, briefly.
Briefly. Thank you very much, Richard. So you made admirable case for life in the universe, but as for the other hypotheses, I'm not too convinced. But having said that, you know, are you proponent of water necessary for life? No other solvent would do, as some scientists do put forward silly ideas like formamide or ammonia.
Actually, there are so many chemicals that require water as a solvent that I think that it is a fairly safe bet to say that water is an absolute requirement for life. All organisms that we know on Earth have an absolute requirement for water in order for life to exist. And also the fact that water is extremely abundant throughout the universe on other planets, exoplanets, and so forth. In fact, water has been found just about everywhere we look. So I don't really think there's a necessity for one to look for other solvents, since water works perfectly. But formamide and other reactions, as they've shown at the Joint Institute of Nuclear Research, can be profoundly important for the origin of life. This is a great mystery that I haven't attempted to solve. I'm only saying that I think the evidence is that life on Earth didn't originate on Earth, but could well have originated somewhere else and been transferred to Earth, not only in something like lithopanspermia, but in aquapanspermia, living in oceans, and the possibility of entire biospheres being transferred intact by a rogue planet coming from another solar system or even another galaxy colliding with the planet Earth at some point in distant past, or breaking up and releasing cometary material that brought life into the ancient oceans of Earth. And that is somewhat supported by the fact that life appears on Earth virtually as soon as you have liquid water oceans, which I think indicates we got infected rather than there was a de novo origin of life on Earth.
All right, thanks, Richard.
Thank you very much, Richard. So we go to the next speaker. It's Andrei Rubin.
Sorry, do we have time for one more question?
Okay, very quickly, please. Yeah.
Okay, thank you very much for so much interesting presentation. My question is, in recent years we have at least 2 interstellar objects that were observed, and they came to the solar system, at least one comet and one asteroid. And there is high possibility that we can see such objects in near future. Is it important to study such objects in future, near future, and to try to find any forms of life on such objects?
Oh, I think definitely, because Oumuamua and 2I Borisov were definitely interstellar material, and it is clear that interstellar comets are coming in, probably much more frequently than we believe possible, because they're so difficult to observe. If we could capture evidence of one sufficiently in time that it would be possible to launch a probe to catch it, it would be profoundly important to do sampling just as was done on Ryugu and Bennu, to look for organics and possibly even biology.
Thank you. Sorry, we should go further to the next speaker. It's Andrei Rubin with his talk strategy for using fluorimetric methods in the search for extraterrestrial life forms.
Okay, well, thank you very much for the opportunity which I have to present our data, which we have developed together with the Professor Skladny from the microbiome. Now, the previous speaker, Dr. Kulmer, made it much easier. My problem is much easier. It facilitates quite a lot by presenting a lot of different data concerning the abiogenetic synthesis of organic compounds which precede the origin of real living systems. What I want to emphasize and to make the first point as a starting for the subsequent presentation is that the existence of organic compounds, no matter how complicated and complex they are, still are not the sufficient condition for the existence of life. It's only the necessary condition, which is manifested now, a very, well, I would say, brilliant manifestation of different compounds which are present in star, clouds, whatever, in the universe. The same story happened accordingly on the Earth. At the early stage of development of our planet, there was the source of energy provided by ultraviolet lights, lightning, all things like that. Without oxygen, ultraviolet penetrated deeply into the atmosphere, and that was the source of abiogenetic synthesis of many compounds. Direct experiments, we all know it's about, in the last century, show that it's really the case. They are formed there. And so, how the origin of life could be well provided, and what are the steps of generation of real living systems, which is an open system, which exchange of matter and energy with the surrounding, and to do that, it has internal metabolic processes and also the limits and the boundary which separates this living system from the outside environment. Otherwise, it wouldn't be a definite system, you know. What I'm saying is just a general point which doesn't need any proof, but it's the starting point for the different methods. And actually, we present two types of methods using using optic apparatus as a source of extraterrestrial life forms. Well, to be more precise, to prove that any parameters which were supposed to characterize living systems should be evaluated with respect to the role of the metabolic processes, which are real proof of life as it is, as a phenomena, not as a presence of some compounds, but metabolic process that are crucial for the as a proof of the existence of some extraterrestrial forms. Now, on the first slide, focus. This one, yeah. What that was? Okay. Okay, here what we see, the first set of experiments, which was suggested by Professor Skladnev, it's that it is well known that cations of organic atoms here can form spontaneously different aggregates and nanoclusters, nanoparticles, if they are reduced by some extra reducing agent. So here is on the first slide, you see that is spectra of the size of those organic nanoparticles. They're uniformly distributed in the environment. But if you see, they have certain spectra, absorbance and fluorescence spectra. But if the same story, the same pattern, we want to observe in the presence of living system, putting those argentum atoms into the living system suspension, then the size distribution is different. There's predominant sizes of those nanoparticles which happened in the presence of those living system of cells. See, I don't know what's matter. I push it. I have to push two times, up and down. Up and that's right, but I didn't. It failed, you know. Okay, so okay, now I found out. Okay, the next slide shows you that what happens if we see these particles at more amplification in the presence of bacteria. Well, this is the Latin name for it. It doesn't matter. Well, anyway, so the argentum, clusters are scattered in the medium. The source of their reduction may come from the metabolic metabolites released by those cells, and make those clusters to form outside of the cells. But what about those clusters with the predominant shapes? They're formed on the surface of the cell membrane, outside membrane, and they are stick to it because on the outside of the membrane, as well known in biology, the negative charge, which is the result of the metabolic process inside of the cell, is the source of those argentum ions reduction. And here is the spectra of the absorption spectra of those particles and the shift of the spectra and dynamics of its formation in time. So this you can also detect that by fluorescence also. And let's see. Okay. Давайте посмотрим, да. Вот это, да? Вот на это. I'm sorry. Ага, вот этого, да? Значит, это спектр поглощения, максимум, он идет в формированию, значит, 40 минут, 20 минут, ну, или так, наоборот, 5, 10, да, динамика, да. И он, да, вот амплитуда их все больше и больше, а кроме того, немножко сдвигаются даже. Ну, well, okay, I'm sorry about that. Okay, I'll speak a bit Russian, but okay. Okay, so it shows you, I'm not going to dwell into many details about the, structure of those nanoparticles, what they are and all that. That's another story. But the thing is that they are, just that the result of metabolic processes. And so what that means is that it is the good method to detect not only extraterrestrial possible mechanisms, but also it's have a very, good, application on Earth as a detection of some organisms present in any type of medium, maybe the source of some toxin, whatever it is, okay. And next slide shows you what happens on the, what are the proofs of metabolism being the source of these nanoparticles and as a result of a reducing agent released from the cells. Well, here is the chloramphenicol, which is the inhibitor of the protein synthesis, is applied to the surface to those cells, and part of this cell colony were not exposed to the effect of this chloramphenicol, whereas the other part was exposed. So the difference is very visible and it's obvious. There is those particles, those clusters, and there is no particles here where amphenicol didn't have its effect. So this is, again, what the point I'm making is we have to be sure that whatever we see as the information coming us from some methods applied for the source of extraterrestrial life is the result of metabolic process, not some chemistry, whatever, which very well can proceed in the presence of those abiogenically synthesized complicated and complex in structure organic compounds. Okay. The next slide shows that another type of, another type of toxins applied to damage metabolic processes. I'm not going into details of those metabolic processes, but since in this case what we made is the toxin which damaged the DNA replication, and as a result it has no effect on the reduction of argentic ions. And so that's another way that there is some process which are responsible for the generation of a negative charge on the surface of on the outside surface of these cells. The other process has nothing to do with it. Nevertheless, and on the other hand, they don't stop metabolic processes in general. That's one way. I didn't prepare a lot of science as a background for this and for these interpretations, but another set of methods we use, optic methods as a search for extraterrestrial life, is the methods we use to detect photosynthetic organisms on Earth or on extraterrestrial, whatever, some surface planets, all that. The basic idea here is this: that light energy played a very important role in the origin of life. That's the energy source of our abiogenic synthesis of organic compounds. And after that, after this happy life with a heterotrophic life, where there was a lot of abiogenically synthesized source of food, this source was exhausted. Photosynthesis came to help to maintain life by providing photosynthetic processes, where the source is light energy, and compounds CO2 and water are the inexhaustible source of matter to provide these things. But how to differ between the simple photochemistry, photochemical, photophysics reaction, which consume electronic excited state energy, singlet or triplet doesn't matter in this case, and how to differ it from those photosynthetic processes which develop in a well-organized living systems. Well, but here is the scheme of photosynthetic process. The main point here is this: after light excitation, electron flow is generated in the complicated membrane sequence of protein electron carriers, and that is the result is the reduction of CO2 by electron coming from the water after it is splitted, and the release of free oxygen is the byproduct of the photosynthesis. By the way, it's interesting, you know, data that it took about 500 millions of years to saturate our atmosphere with the free oxygen, which we all use for breathing. Now, this light excitation has some losses in fluorescence, about, let's say, about 1–2% of fluorescence. And those fluorescence losses can be detected immediately in the solution, that has nothing to do with life. The steady state level of fluorescence coming from the excited states in simple photochemical reactions is established at the time which is about 10 times higher than the average average fluorescence decay lifetime. Whereas what happens, and that's well known to physics dealing with the fluorescence, well-known fact. But what we have here, if we see this curve of fluorescence, how is it established after the onset of illumination of a living organism? Yeah. You see, this is what happens here. I should be very careful, you know, because moving in this arrow in the wrong direction. Here it is, my arrow. Okay, here is the transient curve from the initial level of fluorescence. It's about the sensitivity of these methods are very high. We can detect, getting a little bit ahead, we can detect algae in the water-type water, which we all use for drinking. Now, the transient curve, or they say induction fluorescence curve of chlorophyll, is here. If I find my arrow, okay, here it is. Okay. It has different transient, you know, tops and deeps, and dips. And the time is here about, in millisecond, takes about around 1.5 seconds to reach the maximum level. Now, if we add some toxin here to stop the block electron flow, which is generated as a main product of light consumption. What we have here is that the electrons don't have any place to go, any free room to leave the excited chlorophyll. And so what they have to do to come back, and they come back immediately as compared to the time they spend to go along the electron pathways. So you see the difference again. We block the metabolism, we change the information coming from these living systems. And well, generally, if we make some heating up to 60 degrees, which is nothing exotic here, but we stop metabolic processes without damaging considerably all this machinery, protein machinery, which is responsible for this science. We have something, the transient curve is that what we have in simple solution. no metabolic process, still fluorescence, still life absorption, still chlorophyll. All these organic compounds are present here. They're active, but they don't give this phenomena, which is the I would say the necessary parameter which characterize developed form of life, developing at different stages. It could take 2, 3, no more steps. In this case, it's about, I don't know, many steps here, more than 10 steps here. But we can have 2, 3 steps more than a simple single act photochemical reaction. So that's what we suggest to do as what is the readiness? The project is on the go, so to speak. But what I will say, we developed such methods already applied for the Sputnik, for the satellite observation in the real-time mode. We put our algae on Sputnik, supplied with these apparatus, automatic apparatus. Next presentation by Dr. Konica will clarify it in more details and have information what happened during the flight, not after they come back to the Earth. We spend some time to find them. They're exposed whatever conditions unfavorable for the existence they may eat, and then that's the very, indirect method to evaluate what happened with them during the space flight. In this case, we use this method to detect, not only to detect life, but also to see what's the state of the living systems exposed to different space factors, any type, And so we make the project with the magnetic field effects, things like that, and the first results show that the energy conversion process in algae were really damaged, and we know the mechanism of this damage. And the last point I want to make is that I will say some words about this picture. Is that why to develop astrobiological methods? Not only methods, but research are very important from both basic, also and applied point of view. Well, in case of applied point of view, we developed in combination with solving dealing with autobiological problems. We developed methods such as I said here to apply them for ecological monitoring. Another thing that the basic problem here is what happened to those abiogenically synthesized compounds, what directed them towards the formation and arrangement in a special structure, which we have now as the basic molecular structures, which as the biological base for metabolic processes. If you look on this evolutionary chain from cells to develop life, here it is. The difference is dramatic. But on the other hand, where is my? Okay. But the molecular structure of basic compounds, ATP-F and ATP synthase or ATPase, which hydrolyze ATP, or mitochondrial ribosome, whatever, they are almost the same. They didn't undergo some dramatic changes with respect to the molecular structure. So the basic question is this: what was the driving force for the abiogenic construction at the early stage of biological evolution, or even before the onset of biological evolution, to construct such molecular motors which are ready to satisfy demands coming from the organisms which have been waiting for millions of years to generate themselves on our planet. So that's something to predict, something which you should need in billion of years, and to make it a construction which doesn't need any dramatic changes, that's the crucial, from my understanding, and of course it's limited understanding, it's just a basic question which says, to my mind, is that the mysteries of chaotic behavior, what I mean by chaotic is the classical physical chemistry says that there is such kinetic which undergoes some well-established thermodynamic and kinetic laws. That's okay, but there is some mysteries here which is maxed in the elementary motions, mobility of certain particles, which biology or some pre-biological forms of pre-life extracted and use it for biological purposes. So the existence, the origin of life, generational living system is not the exception, as we understand all of us, but the justified and vectorial result of primary mobility or locomotions of simple particles, elements of construction of this biological molecular motors. Well, thank you for your attention. That's my.
Okay, that's nice. Yeah.
Thank you.
Thank you very much for your very interesting presentation, a new method. We have time for short questions.
No questions here.
Okay.
No question. Maybe we will have some more questions after the second talk, yeah, because it's a kind of continuation.
Okay, yeah. During
okay,
break,
thank you very much. So the last talk will be given by
Maybe
Ivan.
we should have a coffee break, no?
Using colorimetric methods to search for extraterrestrial photosynthetic organisms.
Dear colleagues, nice to meet you. I will focus on the photosynthetic organisms. Photosynthesis is the main energetical processes for life on Earth. Of course, we don't know a priori about photosynthetic organism or its presence on space bodies, planets or asteroids. But what we can try to do is to try to find the same signs of photosynthetic organisms in space. The first step, easiest step we can do is to detect fluorescent molecules in space, because every photosynthetic organism contain pigments, special molecules which effectively absorb visible light under a low concentration of pigments. And one of the famous pigment is chlorophyll. I hope you remember this pigment from our secondary school program, the green pigment. It absorbs blue light, red light, and it also emits fluorescence at the red region of spectrum. To excite this fluorescence, we apply simple devices. One of these devices I built by these hands. Here it is. It's a device made for students, for public presentations, of course. And what's inside? Inside you can see a light source, laser diode or light emitting diode. Then the collimating optics, which generates parallel beam. Some optics to collect outcoming radiation or incoming radiation optical filters to restrict spectral range, to decrease noises, optoelectrical converter, photodiode or PMT, digital signaling processor which converts electrical signals to digital form and that transmits information or data to the Earth. The next step is to recording the fluorescence induction curve, this kind of curve. To do this, the device generates strong flashes, strong black flashes. I hope you see them. Under this intensive light, photosynthesis saturates and the intensity of fluorescence go higher and higher. The duration of this pulse, typical duration, is around 1 second. 1 second is enough to completely saturate photosynthesis. Under these conditions, the energy losses into the heat and into the fluorescence increases. So we can monitor this process. In a non-living system, the signal is very simple. It's just horizontal line. This simple we can find when the chlorophyll is in solution. Or we can take the real biological object, living object, put them, treat it by heating, for example, and this dyed object also will demonstrate the very simple shape of the signal. We called this curve fluorescence transients or fluorescence induction curve. Different kind of organisms demonstrate very close shape of induction curve. The main features are the 2 or 3 intermediate steps. One step in a millisecond range, the second step in tenths milliseconds, and the final stage. What types of objects? Leaf, lichen, microalgae, and macroalgae and microalgae. So cyanobacteria also can demonstrate the fluorescence induction, and also we can measure signal from single cells. Here Professor Pogasyan records fluorescence signals from single cell. You can also see the typical shape of the first component, the second component, and we never see such a complex signals in unliving systems. That's important. So the second step of study photosynthesis in space is the recording the fluorescence transient. But we can also do more. We can apply additional treatment. So we can change the physicochemical factors. We can change temperature, we can change humidity, or we can change light intensity, which we use to excite fluorescence. And all these events will stimulate the change in the shape of the induction curve. This picture shows how the shape of the fluorescence induction changes under the change in excitation light intensity. Low light excitation intensity and high light excitation intensity, different shapes. We also can organize long-term fluorescence monitoring. Now this device generates a sequence of pulses. One pulse, the first pulse, the second, the third, and so on and so on. And we can put this device on the surface, focus it on the object, and try to record long-term monitoring, long-term data. We do this when we study photosynthesis in photobioreactors, for example. Here you can see a vessel, and inside this vessel, chlorella culture is growing, and this fluorimeter continuously measures the number of cells, the amount of chlorophyll inside this culture, and you can see that this red line shows the real dynamic of culture growth. I think the time of this graph is equal to 1 day. So we see how the culture is growing exactly. And we can also record the same process in space. If we see that our fluorescence signals is growing and growing and growing, I think it is a very strong evidence that there is a living system, growing system. Thank you for your attention, and I can just summarize. So we can apply different steps to find out about the presence of life in space. The first step is very simple, is the detection of the presence of the fluorescent molecules, the first easy step. The next thing that we can measure is to reveal the presence of variable fluorescence signals, the complex shape of these signals. It's the sign of the photochemical events inside the system. And the third thing that we can do, we can modify external factors, temperature, humidity, and so on and so on. And if we see that our system demonstrate reactions, certain reactions according to the change in these factors, that we can say that possibly this is a real living system. Thank you.
Thank you.
Thank you very much, Ivan. Actually, very interesting. Two very interesting talks about the method important for astrobiological studies. Actually, we have a kind of astrobiological group who are dealing with implementation of methods for detection of life, and it would be nice if you could join us for some seminars and for some discussions. Thank you very much. So, Stohan, you have question?
And we also have a question. Can we?
Okay.
Who first?
Who is the first? The audience has question?
Okay, I have a very short question. Do I understand correctly that at this plot it's on the screen, the final level of fluorescence does not depend on the initiation intensity level? What determines this final amplitude?
Final amplitude is determined by the total amount of chlorophyll, but also it depends on the organism. Chlorella cell will give you one curve, but lichen body will give you another shape of this curve. But it's about the details. But the typical shape of this curve, multi-step and lifetimes are typical and close among different living organisms.
Okay, thank you. We have another question in the audience.
Yes, please.
Yes, I'm very much interested in this. It's a wonderful presentation. Can you distinguish by the fluorescence response between different types of phytoplankton? For example, can you distinguish between diatoms and cyanobacteria and dinoflagellates by their fluorescence response characteristics?
Thank you. We can distinguish between eukaryotic and prokaryotic cyanobacteria photosynthesis. Look at this picture. There is a lichen which contains cyanobacteria inside. And cyanobacteria has a typical time needed to reach the maximum around 1 second. But when we measure the signal from green algae, like chlorella, or diatom, the time that we need to achieve to reach the maximum is shorter times, 100 milliseconds. So we can distinguish eukaryotic and prokaryotic photosynthesis. Looking at this.
There's a question from the audience.
Okay, how universal is this method? Is it related only to a specific kind of bacteria or living systems?
It's wondering how
it is related to
how universe is this method. Here I demonstrated different types of photosynthetic organisms, and all of them demonstrate this complex induction phenomenon. There are differences in details. So one organism has 1 millisecond component, another 2 millisecond component, but all of them will generate the first rise in the millisecond range, the second rise in the 10 millisecond range, and the third around 100 milliseconds.
Thank you. Sohan, please, your question.
Yeah, thank you very much. My question is that how far will you be able to see, inverted commas, how far will you be able to see or detect life? Is it 600 light years, or at the end of the Milky Way galaxy, or within Andromeda galaxy? How far can you go?
Thank you. Good question. Do you mean how far distance? Yes, distance.
Yeah, distance, light years. Obviously, it's not going to be kilometers.
Okay. To induce this fluorescence rise, we must provide the high level of optical power density, not total power, but power density. So we must apply collimated beams to induce fluorescence rise. And in practice on Earth, I know that the systems which can excite fluorescence from the distance around 100 meters are built. 100 meters, maybe 1 kilometer. So it depends on the sensitivity of a telescope. It depends on the diameter of the collecting lens or mirror, so the quality of the telescope.
Right, okay, thanks.
Thank you. More questions?
No, we don't have any questions.
No, so thank you very much. So the session is closed, and now we have a coffee break. There is a suggestion for 10 minutes here for coffee break, and then we can continue with the poster session. Is it okay?
Yes, good. So let's meet again in 10 minutes.
And we have 10 minutes coffee break.
Thank you so much, Oleg. It was wonderful to be able to join you today. Thank you.
Thank you. Thank you, Richard. Always nice to see you. Next time, face to face, I hope. Yeah.
Hopefully, I will be able to join you next time. I missed coming to the Moscow Symposium. It's been wonderful being part of this program. Thank you so much.
Welcome. Bye-bye. Yeah, thank you. Bye.
Bye-bye.
Bye, Richard. Oops, it's gone.
No, I'm not gone. I just I just turned off my video. I'm still here. But nice being with you. Thank you. Bye-bye.
So should we start our poster session?
We are still waiting for colleagues coming from coffee break. I propose to wait for 1-2 minutes.
Yeah, no problem.
Okay, we can start.
We can start, yeah, please. So it's Marina Greenberg.
Yes.
With her poster, please.
Yeah, we don't have questions in poster session.
Okay.
It's a reminder.
Good afternoon, dear colleagues. I will talk about the effects of ionizing radiation and magnetic fields on plants. Why is this important to study? Currently, projects for long-term space mission are being developed in Russia and around the world, at orbital station, on the Moon, on Mars. To support long-term autonomic mission, artificial ecosystems are needed. We, biologists, must be prepared for this. Why ionizing radiation and magnetic fields? These factors vary little on Earth but are greatly modified in space. The reaction of living organisms to such change have been poorly studied. Why plants? Plants are the primary producer of an ecosystem. Food and atmospheric regeneration depend on them. What parameters to study? The most important of them is growth rate, activity of physiological processes, and status of signaling system. Today I will only talk about photosynthesis, which is a key process in plants, and about the signaling system that are responsible for the participation of environmental factors. The first factor is ionizing radiation. The experiments were carried out on wheat plants. Plants were irradiated using a closed point source, which provides the possibility of exposure through the entire period of plant growth. The dose rate is 200 times higher than the natural background on the Earth and approximately corresponds to the condition on Moon and Mars. On this slide, you can see an infrared gas analyzer and a pump fluorometer. This setup can measure the activity of different stages of photosynthesis. Our experiments show that ionizing radiation in low doses cause weak stimulation of photosynthesis. Like human and animals, plants have a stress signal system based on the transmission of electrical impulses. Having arisen in the stimulation zone, for example, as here at the heighted tip of the leaf, such impulses spread throughout all the plant, and we can record the pulse parameters using special electrodes. We found that ionizing radiation in low doses causes a significant increase in plant electrical signals. What about magnetic fields? Research had also been conducted on wheat plants. We changed electromagnetic environment was set by Helmholtz coils. In this set of experiment, we haven't not directly simulation space conditions, but rather studied the general effect of magnetic fields. Similar to ionizing radiation, low-intensity magnetic fields have virtually no effect on photosynthetic activity of plants. In the figure, you can see a spectacular display of different photosynthetic parameters, but there is practically no difference between the control and magnetic fields. What about electrical reaction? Yes, the effect was found for them. The experiment records the electrical reaction of plants in response to switch on of light. Low intensity magnetic fields cause a significant increase in plant light-induced electrical reaction. The effect on signaling system discovered during the work indicates that plant exposure to altered level of ionizing radiation and magnetic fields may adapt differently to additional environmental stress factors. Such impacts may also occur in space greenhouse conditions. And conclusions. Theoretical analysis was shown that one of the most important factors of spaceflight that can influence the state of plants is altered level of ionizing radiation and the environmental electromagnetic environment. No statistically significant effect of plant growth were found for either ionizing radiation or magnetic fields. A weak stimulation effect of both factors on the key indicators of photosynthesis, the quantum yield of photosystem 2, was found. A significantly more pronounced effect was shown for signaling system. Ionizing radiation enhance heat-induced electrical signals, and magnetic fields increase the amplitude of light-induced electrical reaction. In general, the results indicate that already low intensity of space flight factors can affect the development of plants, in particular, their ability to form adaptation to stress conditions. Thank you for your attention.
Thank you very much. So we have two more speakers, but I don't know if they are online.
We have the first one. We have Daniil Barbashin.
Okay, Daniel.
Yes, Daniil, please.
Good evening, dear colleagues. My name is Daniil Barbashin. Today I would like to present the results of our study named Bacterial Tolerance to the Influence of Sodium Perchlorate Estimation in Extreme Ecotopes Communities. The relevance of my work is due to discovery in 2009 by the Phoenix mission of perchlorate salts in concentration of 0.4 to 0.6% in the regolith of mass. These salts have interesting properties, which make it possible to concentrate water from the atmosphere as a liquid under mass conditions. All of the above indicates a new potential niche for microorganisms to live in the condition of the red planet. Also, in recent years, microorganisms have been described that are resistant to high concentration of perchlorates. However, the most stable bacterial strains have been discovered over the past years. For example, in 2017, the most resistant bacterium currently known was discovered, Planococcus gallocreophilus. In general, the maximum concentration of perchlorate that limits the vital activity of microorganisms had not yet been established, which poses the question to the scientific community: is it possible for microorganisms to survive at high concentration of perchlorate capable of ensuring the existence of liquid water in the max conditions? So, during the study, we tested 5,445 strains from 51 soil samples. Sedimentary samples were taken from 18 regions from different conditions: hot and cold, arid, saline, and others. In our work, we used the next methods, which shows on this slide. 5,445 isolates were analyzed for resistance to the presence of 7.5% sodium perchlorate in the medium. The most stable communities were communities from ecotopes associated with tolerant habitats. There are White Sea coast, coast of Lake Boskonchak, Sarpinskaya lowland. Next come 4 communities with resistant from 6.5 and 8%. 800 strains isolated from soils in 6 regions were tested for resistance to 10% sodium perchlorate. The largest shares of resistance culture are Hrinovskoy pine forest, followed by Sorpinska loveland and others. Some strains of genera Nocardia, Pseudomonas, Bacillus, Rhodococcus, Artrobacter, Massilia, Micrococcus showed resistance to the presence of 10% perchlorate in the medium. All of the genera listed have at least several strains adapted to the effect one or more extreme factor. 4 strains of the genera Kotsuria, Atrobacter, Micrococcus, Bacillia, Pseudomonas, at least one species capable of perchlorate reduction is described in the literature. According to the literature, one bacterial strain is superior in resistance to the discoverer. It's Planococcus gallocreophilus. It is also worth noting that the above microorganisms are among the most resistant to perchlorate that have been described so far. Thus, as a result of analysis of 5,445 isolates, the occurrence of strains resistant to 7.5 and 10% of sodium perchlorate was 3.4% and 1.3%, respectively. Bacteria capable of growth in the presence of 7.5 and 10% sodium perchlorate were found in 83 and 67% of study ecotopes, respectively. The large proportion of resistant strains were identified among culture isolated from soil samples of Surpinska lowland and the coast of Boskonchak. Cultures capable of growing on the medium containing 10% sodium perchlorate were discovered. This was strain of genera Nocardia, Pseudomonas, Bacillus, Rhodococcus, Arthrobacter, Massilia, Micrococcus. Bacteria of genera Kotsuria and Bacillus were found capable of growth in the presence of 20.5% of sodium perchlorate. Thank you for your attention.
Thank you, Daniil. Very interesting study about perchlorates. It's always interesting in relation to Mars, especially. Do we have anybody to continue our poster session?
Let's ask, is Mehdi Hodadardilori?
But I think he is not here, yeah?
Yes, he's not here. Alexander Safronov. Okay,
No.
so these two people, they are not here.
Okay, so it looks like that we are finishing exactly in time. Okay, so then I can only say thank you very much for the organizers. It's always very interesting conference for me and astrobiology section also. So if somebody could like to say something, that would be nice, yeah. Yeah, you can say something, yes, Johan.
Sorry, what was the question?
Yes?
What was the question? I missed.
Which?
What is the question? Would you asked us a question?
Which one?
You say you'd like somebody to say something.
Yes, yeah. So simply you can say something if you want at the end of the conference, because we are closing already, about to close.
Well, I'd like to thank the organizer, Alexander, I think his name is. Is it Professor? Who is the organizer of this conference? What's his name? I know his name. I think it's Alexander, isn't it?
So you mean the which organizers? The head of the conference or the team who is organizing? Because
the head of
yeah, the head of the conference is Lev Matveyevich Zelenyy. Yeah.
No, I am just chair of program committee, but there are many people involved, and this session very interesting day-to-day organized by Oleg Kurtsubenko. It was very interesting session. I am not specialist in astrobiology, but I have learned a lot. It was really very interesting. And I want to thank all speakers. And as we discussed with Oleg briefly, but we think about making this type of seminars regular. And Oleg, we will get in touch and we ask you to convene it. It's possible to do from abroad. But if you want to make people in Russia interested in astrobiology, get them together. This is, I think, the best beginning. Have seminar, and people will concentrate about it, and then we could think later about some development and some program. So, Oleg, if you agree, we should think about details.
Yes,
Well,
yes, I agree. Thank you.
I'm going to investigate whether the British government will hinder me to coming to Russia. I'll come. I don't mind traveling to Russia if it's at all possible, because I'm so determined to put forward this education program across Africa. We have the opportunity to do it. I have the universities working with me. And one of the problems, for example, I'm encountering, which I sorted out today, to be quite honest, which is that for me to ship any apparatus out to Africa, I would need a permission from the British government. And that, for example, is going to take 3 months. And once we got the permission to do that, because it's going outside the European Union, people tell me, you can't just do it. And after 3 months, I think it'll take 6 weeks to make the apparatus, put all the packages together. All right, we're friends here, say 2 months. So that'll take about 5 months. And then obviously I have to go to African universities and put it together, rig it up and make it work. So it'll be at least 6 months by which time I will do it. I will be able to have a working program in Africa. And where we are so far, as I said, we're in the process of putting this, I'm in the process of organizing this making of the apparatus. I'm in the process now. And the other thing is that we have got astrobiology program which kindly our friend Oleg put together. I put together the astrochemistry and astroscience, and then we have astrophysics, which I'm going to do the astrophysics, and I'm only going to do one unit because I'm not astrophysicist. I'm only going to talk about the energies. I'm glad somebody asked me a question about the energies. So I'm going to actually talk about that. I'm not going to talk about deep astrophysics, like, for example, what are alpha rays. You know, I don't know what alpha rays are. I don't think anybody knows what the alpha rays are, you know. And things like these, although it has not, I think they use alpha ray more than the classic physics sense, you know, alpha beta ray, you know, rather, I think they use it more than that because they're the highest energies in space. And I mean, that kind of kills anything, it destroys anything and everything in its path. And you, sometimes I wonder, well, if they are that strong, you know, so why are the dark molecular clouds exist at all? Because they should be incinerated. Because anything beyond X-ray, you can knock out in addition to the valency electron, the rest of the electrons as well. So the X-rays are too high energy, and alpha rays fall far beyond that. So I have no answer for that. But what I do have an answer for, actually, the high energy particles, and I can write about those kind of things, and that would be my input from the astrophysics point of view. So we've done quite a lot of work on this, and I'm just about to hold a meeting.
Dear Sohan, I have suggestion. Sorry to interrupt you, but it's late and just people are waiting. We have many specialists in astrophysics in our institute, and for us, travel to Africa is very easy, much better, easier now than to Paris. So if you invite them, we could send you people specialists in high energy astrophysics, in X-ray, in gamma rays, and cosmic rays, and they could read your lectures. But I suggest better. My teacher, Professor Sagdeev, said it's good to speak about love, but if you want to discuss business, it's better to write. So please write me a letter with your suggestion for cooperation and how we could help, and we will be happy to discuss it.
Thank you very much. Actually, you're a godsend, as we say in Britain. In Britain.
Sorry, maybe I missed what particular city in Africa you have in mind. Where you're working? What university?
Well, what I have in mind, as I said earlier on, I mentioned 4 universities. We're going to begin with the Nigerian University at Nsukka, as well as with the Nigerian Space Agency. Collectively, both of them together, we'll be communicating with them, and we want to deliver lectures in Africa. We want to teach the lecturers to teach their people.
It's okay. Nigeria is a very good country for us because President of Russia is a good friend of President of Nigeria. They recently met and really was very friendly. So
you know that you made me happy because actually you could take out one of your colleagues can write the astrophysics for the beginners, sort of first year student, so they could actually write the program, and we can actually work on that and then deliver some talks so that
it's possible. Just I think we need budget for it. I think it's maybe your university we could invite these people for say 5-6 lectures. We find some good specialists.
No, we can't. We can't. You have to fit in the framework of what they told me to do. So I can fit in. I can fit in two astrophysics, 2 1-hour lectures, and then we need 2 of the astrophysics. Astrochemistry. We need. Of course, Oleg was eager, and he did 3, so that's 7 altogether. And then we have, I have a colleague who wants to actually.
Okay, let's discuss it between us, not to have other people waiting. Thank you. Please write me, and we try to do as much as we can.
All right. Okay. Look.
Okay.
One thing, as I said to the Western world, which aren't listening, and we can work together, and we can put this program together, and that's all I'm trying to say. We'll work together on this, I welcome Russian input, and of course you have access to the university, to the space agencies, everything else. And then we move on to Ghana, then we move on to Kenya, and then we moved on to Zambia, And
no, this is too much. Only one country.
It's not too much. The way that I have it in my mind, it's not, because we're simply going to teach the teachers. We're not actually doing it ourselves. We will be doing ourselves lectures via Zoom, like for example some serious astrophysics calculation will be done by somebody from Russia, some person. In fact, I have another friend who is an astrophysicist. His name is Oleg do you remember Oleg, the other friend I introduced to you once? Do you remember we went for something to eat? Do you remember his surname, Malkovich? I'll give you the name in once. All right, okay, everybody's waiting, but yes, I'll try. I'll communicate with Oleg, and he will communicate with you, and I'll try and come to Russia to talk face to face. Is that okay?
Okay.
Yeah, it's okay, Sokhan. Thank you very much. It's always nice. Your contribution is very valuable.
It's
so
time to...
now I
Oleg, thank you for chairing this interesting session, and thanks all participants. And it's that moment our meeting is over, our session is over, and our 14 conference is over, and we're waiting everybody to 15 conference October next year. But we hope to have communicate through this new seminar if we manage to arrange it.
Yeah.
Goodbye, everybody.
Bye. Goodbye.
Thanks to all. Yeah, thank you.
Thank you.